Hello.
Hello, good morning, sir.
Good morning. Good morning.
It's 9:35. We can start the session, sir.
Okay. Okay. Just meet. So, good morning, all of you. I welcome you all.
Good morning, sir.
Good morning. Good morning. I welcome you all in this faculty development program on techniques for research in chemical and biological sciences, organized by Professor Ramkrishna Murray College in association with UGC, Human Research Development Center, Savitribai Phule, Pune University. Today it's our last day, and we are starting from first session. Up till now, we have lectures on spectroscopic techniques like UV, IR, NMR, also some DNA sequencing, then omics. Today we have session on nanomaterial and about their characterization. So, on behalf of Ramkrishna Murray College, I welcome you all. Today's guest, Dr. Milind Kulkarni, they also join in this session. So, on behalf of Professor Ramkrishna College, I welcome
Hello.
Kulkarni from Summit.
Hello.
Hello.
Good morning, sir.
Good morning.
Hello.
So, hello.
Yes, sir. We can hear you, sir. Yes, sir.
Yeah.
Sir, you are audible,
And
sir.
my screen is visible to all of you?
Yes, sir. Yes, sir.
Yes, sir.
Okay. So
So,
very good morning to one and all.
Sir, Dr. Rasali is going to introduce you, sir.
Yes, no problem. Yes, please.
Okay, sir. Okay,
So,
sir.
Yeah.
I request Dr. Pawar, sir, to introduce our today's speaker.
Good morning, sir. Good morning, Kulkarni, sir.
Very good morning, sir.
Okay. Good morning, one and all. Now I will start the session first from 5th day, FDP of HRDC, conducted by Professor Ramkrishna Murray College, Akodi. Now I introduce today's speaker for the session 1, that is the Dr. Milind V. Kulkarni. He presently working as a senior scientist and group head at a nano composite laboratory, Cement, Pune. Sir completed his MSc degree in polymer chemistry from Shivaji University, Kolhapur. Sir also he has received his PhD on conducting polymer based sensor in 2002 from University of Pune in 2001. He started career as a scientist B at Cement, Pune. His main field of current scientific interest is nanomaterial and polymer composites for multifunctional application. Sir also develop conducting electroactive polymers and their composite in flexible and workable electronic devices. Sir also working in a different field for the applications of this nanomaterial in a different field. Sir published more than 105 research paper in a reputed international journal of high impact factor. He is a Google Scholar citation near H index is 31 with a citation more than 2,963 with a I-10 index 64. He has filed more than 20 patents. Also, he has presented more than 130 papers in a national and international conferences, deliver 50 invited talks. He guided 5 PhD students, 1 more students are current under guidance, and 6 MPhil students. They are sir also affiliated our center as a MPhil guide. He Sir also received GRF, SRF from DSC, CSR, New Delhi, India. He is a recipient of prestigious Young Associate Award in 2009 and a fellow of Maharashtra Academy of Science in the year 2013. He is a life member of Material Research Society of India and to the Society for Polymer Science in India. He is also a board of study member of Ferguson College and also is work DY Patil and he recognized the research guide from the Pune University. He sir also has been invited as adjunct faculty in the Applied Science Department at College of Engineering Pune COP. Also, he is working as an expert for tech novels at Automatic Resource Association Pune. Recently, this is most of the creditable discovery of Dr. Milind Kulkarni. Recently, Dr. Milind Kulkarni has designed and developed the first Indian origin and low cost the polymer swab for the collection of the sample, collection and testing of the COVID-19 sample and other viral diseases. It is my great pleasure to introduce the sir such a huge biodata, but I given only brief informations are only read in this session. Now I request the Dr. Milind Kulkarni please guide our participant, which are the participant in FDP on the techniques used for the nanomaterial characterization.
Yes.
Please, sir.
Yeah. Yes, sir. Thank you, sir, for such a nice introduction about me. And first of all, I would like to thank principal of RMC College as well as coordinator of this FDP for giving me an invitation to deliver an expert talk. Thank you, sir. So a very good morning to one and all. Today I am going to present the research that we have developed on nanomaterials and nanocomposite for advanced multifunctional applications. Okay, so just to begin with, let me introduce about the CMET, that is our institution. Okay, so CMET, that is Center for Materials for Electronics Technology. Okay, we belongs to Ministry of Electronics and Information Technology, that is MIT. Okay, at New Delhi. We have 3 centers located at 3 different geographical locations in India. Okay, in Maharashtra, we have a laboratory and headquarter laboratory in Pune, that is CMET Pune. At Andhra Pradesh, we have other center of our CMET Hyderabad, in Hyderabad. And in Kerala, we have the 3rd center of our CMET, that is CMET Thrissur. And all these 3 laboratories have different core as well as thrust areas to work upon. Okay. So at CMET Pune, we are working on nanomaterial and devices, integrated electronics packaging, then materials for renewable energy. Okay, that means energy storage and energy generation. These are the thrust areas of the CMET Pune laboratory. While at other center at CMET Hyderabad, we are working on ultra high purity materials, particularly metals. That is 5N purity metal, that is 99.999 purity, and these high purity metals or materials are required for our space application, and we are supporting to the space sector through our R&D activity. While at our 3rd Thrissur center, we are working on piezo sensor and actuator as well as ceramic based material, and we have my colleague over there, they are also working on the carbon and aerogel based supercapacitor as an energy storage devices. So this is all about the R&D program at a glance at our 3 centers in CMET Pune, CMET Hyderabad as well as CMET Thrissur. So during my today's presentation, I'm going to give the introduction about the nanomaterials, and then I will introduce you all of you about the polymer nanocomposites, their methods of synthesis, and all the characterization techniques that has been used to characterize this nanomaterial as well as polymer nanocomposite by using various sophisticated analytical techniques. Apart from this, I'm going to introduce you all the applications of this nanomaterial as well as polymer nanocomposite in biological application, particularly antibacterial and antimicrobial activity, then sensor for humidity as well as pH measurement, and we also try to develop the electronic nose for certain volatile organic compounds. Then a very interesting application of this nanomaterial, I'm going to introduce you for the radiation shielding application, that is X-ray shielding application. And lastly, I will present the R&D activities related to energy storage devices, in particular batteries, and that too lithium-ion batteries, which is a very hot topic nowadays. I'm sure you are seeing my presentation clearly, right? Hello?
Yes, sir. Your presentation is visible.
Okay, sir. Yeah. So this is all about the introduction to nanoscience and nanotechnology. But before proceeding, we should know what are the questions to be considered when we are dealing with the nanomaterial, nanocomposite, and their application. So these questions are: what is nanoparticle? How big is nano? I am sure during last 3-4 days, you have been introduced with this nanomaterial, their range, their size, their characterization techniques. But definitely, to just brush up your knowledge, I'm going to present just introductory few slides before coming to actual presentation of R&D activity that we are carrying out at CMET Pune. So what is nanoscience? Why we need nano materials, okay, rather than their bulk counterpart? Then what is nanotechnology and how we can take the opportunities of this nano dimension or nanostructured material for versatile or multifunctional application? And the most important thing is that how or what characterization techniques that we can use to characterize this nanomaterial or nanostructured materials. So just I will start with the nanomaterials. So what is nano? Nano is nothing but a prefix that means very, very small. Okay, we cannot see the nanomaterials by our naked eyes. And the nanomaterial is the 1 billionth of a meter. That is 10 to the power minus 9 meter. You can see on the right hand side, it is a million times smaller than the smallest measurement that you can see on our ruler. Okay, it is a millionth of millimeter or billionth of a meter. This is the size of a nanostructured or nanomaterials. Then what is nanoscience? Nanoscience is the study of structure and unique properties of a matter at the nanoscale level. Okay, so an interdisciplinary field of science that combines physics, chemistry, biology, biological science, engineering, and related disciplines. Okay, so this nanotechnology or nanomaterials has bring all the these different areas of science and technology under one umbrella. Then nanotechnology is the understanding and control of the matter at the dimension of roughly at around 1 to 100 nanometer, where the unique phenomenon can have the versatile application. So just I will give you why we need these nanomaterials. Because the nanomaterials differ their properties with respect to their bulk counterpart. These materials are highly energetic, they are highly reactive or active. Okay, so just there are some student community as well as some young researchers are there. So I will just briefly explain the classification of this nanomaterial. We all know that bulk particle have size in the microns region, while submicron particle size having the dimension of around 100 to 500 nanometer. The nanoparticles are have the size of around 10 to 100 nanometer, while the materials having the size of around 1 to 10 nanometer used to called as a quantum dots. Okay, these are a very fine and minute particles nano of the material. So how big is the nano? Here on the screen you can see the slide or picture. The ratio of earth to football is equal to the ratio of football to the fullerene. So by seeing this slide, you can compare how is the size of the fullerene, that is carbon-60 molecule that appear as compared to your earth size. So what is a nanoparticle? It is the well-known example is the buckyball or the carbon-60. Okay, they are linked together in a one unit and its size is around roughly 1 nanometer. So what is a nanoparticle? Their origin, their chemical composition. These nanomaterials can origin from the natural source, we can unintentionally, they will get released and we can manufacture at laboratory or industries. Okay, they have the chemical composition ranging from metal oxides, polymer, carbon semiconductors, biomolecules and compounds, so on and so forth. They can disperse very easily into a gases in the form of aerosols. They can get dispersed very well into liquids as a ferrofluids and into solid as a matrix material. And there are different structure of this nanomaterial like nanocapsules, quantum dots, ultrafine aerosols, then nanoparticulates material, nanoparticles, nanotubes, so on and so forth. And they have different shape or structures like spheres, needles, platelets and tube, and we can characterize this material, we can seize this morphology or shapes of this material by using various sophisticated techniques like microscope, then FECM analysis and transmission electron microscope. They can aggregate as a single particle or aggregates or agglomerates, and we can have a surface modification by using a polymer and other materials. So I will give a very well-known example of this a nanostructure material and bulk material. We are using gold in the form of an ornament. Okay, we can have a ring of golden that we are wearing into our fingers, and bangles, particularly ladies, they are wearing it. So the bulk gold is in yellow color, but when we convert this material into a nanostructure material, it appears as a red in color. Okay, so the particles are so small that the electrons are not free to move about as in a bulk gold. Okay, because of this movement is restricted, the particles react differently with the light. Okay, whenever light fall on it, they can react in different way. So here on right hand side, you can see 12 nanometer gold cluster of the particle look like a gray or brown in color. Okay, while the bulk gold color is yellow in color. So depending on the particle size, they reflect the light and absorb the light in a different way. Okay, so what exactly happened and what changes will take place when we transform the bulk material into nanostructure material? So the optical properties, crystal state, then conductivity, magnetic properties, melting point, chemical reactivity, so on and so forth, even mechanical strength, surface properties will get changed or get altered when we change the material from bulk to nanostructure material. So what is nanoscience? Already I have given the introduction about the nanoscience. It is the study and manipulation of the materials at the nanoscale level. So why we need that we have to convert this bulk material? We are working into our day-to-day application. So what is there that we have to convert this bulk material into nanostructure material? Why it is necessary? So the reason behind that is at the nanoscale, strange thing happened to the material. That means their properties can drastically change. Okay, and what particular properties? That is reactivity. As particles get smaller, they tend to react differently with the environment than the large or bulk particle. The size, the smaller particles have a different optical properties. Their color changes because the different size of the particle reflect or absorb light differently. So I will give an example of magnetism. The smaller particles can have a different magnetic properties than their larger or bulk counterpart. Hundreds of years ago, our ancient people, they know the art, okay, of preparing and using this nanostructure material. The red stained glass gets its color from the nanoparticle of gold that are around only 20 nanometer across. Here you can see the pictures where the red color is to the structure or painting. It is because of this gold nanoparticles having 20 nanometer size. While the orange glass, okay, orange color glass gets its color from the gold, same material, gold nanoparticle, but that have different nanometer size, that is 80 nanometer across. And now we called it as an nanotechnology. Okay, so what is nanotechnology? Nanotechnology is nothing but the development of material and devices by exploiting the characteristic of a particle or material on the nanoscale. Okay, and this technology is made by humans. Okay, so this is artificial nanotechnology, we can say. So potential impacts of nanotechnology, there are various sectors starting from health, as we know, diagnostic, cancer treatment, because cancer is a very dangerous disease, and nowadays the threatening thing is that it is becoming a very common diseases, okay, all over the world. So to provide relief to the patient, I mean, doctors are trying to develop the material or medicine in a such a way that they have targeted drug delivery and will work particularly to the tumorous part of the, I mean, the cancer species or cells. Yeah. Okay. So the potential impact of nanotechnology, particularly in the material aspect or material perspective, is that we can use this nanostructure material for sport industries, cosmetic industries, clothing, and space elevator. Clothing means we can use different dye molecule nanostructure material to change the shade of the color as well as dyeing ability. Okay. And this is a favorite slide we can say, because we are using mobile phone very often, or rather I can say it has become the organ of our human body, I can say. So this technology of this mobile phone as well as this laptops and computer has been blessed by this nanomaterial, because we have faster processing, okay, morphing of computer, and smaller, okay, miniaturized device that has to be more powerful and mobile devices by the use of nanoscience and nanotechnology. Okay, this nanomaterial and nanotechnology has a potential impact, okay, on the environmental. So we can use this nanomaterial and nanoscience nanotechnology for cleaner energy, better energy storage in the form of supercapacitor, batteries, okay, or fuel cells. And we can use this material for wind energy, clean water for drinking, as well as solar cell to harness the solar energy. So how do we build these small things? Okay, the computer chip can be made from top-down approach. There are two ways to synthesize this nanostructure material. One is your top-down approach and the other one is bottom-up. Okay, so metal nanowires are made by bottom-up. That means that we are building something by assembling a smaller components. Okay, yeah. So like building a car engine or a Lego. Okay. So it is a question of size. This slide, it is very easily available on the Google, and this is a very introductory slide about this nanotechnology and nanomaterial. So I will simply skip this slide. So what techniques we can use to characterize this material beyond the magnifying glass? Okay, magnifying glass we are using to see a very smaller object, because we cannot see it clearly by using our naked eyes. So this nanomaterial we can characterize by using optical microscope, electron microscope, surface analysis, elemental analysis, and many more applications. Okay, there are many techniques for measuring the size of nanoparticle. We can use to differentiate or to classify this size of this material from say about 100 microns to 1000 microns in size. But for a very smaller, that is 0.01 to 0.001 a micron size material, we have to use sophisticated technique like AFM, STM, then electron microscopy, so on and so forth. Okay, so after synthesizing this nanostructure material, we can synthesize them by various techniques like your soft chemistry, then sol-gel process, chemical precipitation, hydrothermal techniques, ball milling, okay, then microwave digestion, spray pyrolysis. There are n number of methods to synthesize this nanostructure material in our laboratory. After synthesizing this nanostructure material, we have to characterize their properties. We have to identify their size and shape. And for that matter, we are using different sophisticated analytical techniques like X-ray diffractometer, that is XRD, and various electron microscope, okay, that is scanning electron microscopy, same for the surface analysis. Then transmission electron microscope, TEM, when we want to see, suppose we have a carbon nanotubes, okay, so by seeing the carbon nanotube, we can see what are the materials that already present inside this carbon nanotube. We have doped it or modified. Just I will go to that particular slide. Yeah. Yes, tunneling microscope we used to call as STM, and atomic force microscope, that is AFM. Under AFM, you have a three-dimensional image or topography of your nanostructure material, that is x-axis, y-axis, as well as z-axis. Okay, so by using STM and AFM, which are useful for surface image technique and can produce a topographic image of the surface of the nanostructure material. It will give the atomic resolution in all the 3 dimensions, as I have mentioned, X, Y, and Z axis. And by combining with appropriately designed attachment, we can see the structure. Okay, by using STM and AFM as a broadened range for different applications like its nano indentation, nano lithography, and we can pattern arrangement or self-assembly of this nanostructure material or nanometals we can do by using these techniques. Almost all solid and surfaces can be studied with STM AFM, whether irrespective they are very hard or soft, whether they are electively conductive or non-conductive. Okay, insulating material also we can characterize and analyze by using STM and AFM techniques. The surface can be studied in air or in vacuum or in liquid state. Okay, so I will give the detailed applications of this characterization techniques. To start with, we will proceed with the X-ray diffraction. So XRD is a very important technique. Okay, and after synthesis of this nanostructure material, it will address the issues related to crystal structure of solid, lattice constant and its geometry, identification of unknown material, because we have the library and we can compare the peak position, their intensity, and we can compare the spectra of our unknown material with the known or the library of this XRD material available with the software, and we can compare and identify the material and its composition. The orientation of single crystal can also be studied by using XRD technique. Preferred orientation of polycrystal, as well as apart from these advantages, we can study the defect and stresses that has been involved in the structures. Now I will proceed with the other area of the material, nanomaterial, that is polymer nanocomposite. Okay, so what is nanocomposite? We have to see what is nanocomposite. Then I will go to the glass and polymer nanocomposite. So nanocomposite is nothing but the incorporation of nanostructured material or nanoparticle in amorphous material or porous material. Okay, the amorphous material or porous material, it may be in the form of glass or polymer, and they have different properties than their host, and the stability of this nanocomposite is more. For example, I can tell you that gold or silver nanoparticle in glass or polymer matrix, to give you glass nanocomposite as well as polymer nanocomposite, we can do metal oxide sulfide nanoparticle in glass as well as polymer, that is amorphous media, to give you different glass and polymer nanocomposite. And metal nanoparticle can disperse very well in the form of a length, width, or thickness, the nanometer size range. Okay, so nanoparticle is nothing but a 3-dimensional structure. Nanosheet is 2-dimensional structure, and nanowire is a 1-dimensional structure. If you take a carbon nanotube and vertically you cut it and flatten it, you will get a 2-dimensional graphene structure. Okay, which has a very good application in energy storage as well as sensor and many applications. Okay, so for making this polymer nanocomposite, you can use different polymer matrices like thermoplastic resins. For example, polystyrene, polyvinyl chloride, polycarbonate, polypropylene, so on and so forth. Then thermoset resin also you can use. For that matter, you can use phenolic resin, epoxy resin, aminos, and so on. And if we require some elastic or flexible application of this polymer nanocomposite, we have a choice to use natural and synthetic rubber or styrene butadiene rubber to prepare this polymer nanocomposite. Under the a category of nanofillers, you can use carbon nanotubes, that is CNT, both multi-walled as well as single-walled carbon nanotube, carbon nanofiller, that is CNFs, metal nanoparticle like silver, gold, cobalt, so on and so forth. You can use Montmorillonite organoclay to prepare this polymer nanocomposite. So what is the different method to prepare the polymer nanocomposite? There are 3 important methods to prepare this nanocomposite. The first is solution processing. Here, the title or name itself will explain what is solution processing. Here, what we are doing is nanomaterial we are dispersing in a very well manner into an adequate solvent in which polymer is soluble. Okay, so here what is the basic requirement is that the polymer must be soluble in that particular solvent. Okay, the next method is melt processing, because there are certain polymers which are not soluble at all in any media, either organic solvent or in aqueous media. So to prepare this polymer nanocomposite by using this insoluble polymer, what we are doing is we have to heat this polymer to its melting temperature so that it will go into a molten state. Okay. Now we have the polymer in a molten state, and we have to add this nanofiller or nanostructure material into the molten state of this polymer. The advantage of this melt processing method is that in this case we don't require any solvent because the organic solvent are very costly. Okay, so without the use of any costly or solvents, we can develop the process to prepare the polymer nanocomposite by using melt processing technique. And the third, which is very important and most practice or commercialized method, is in situ polymerization. Okay, here in earlier two methods, that is solution processing method as well as melt processing, we are using a ready-made polymer material. Okay, so in this in situ polymerization, now we are going to use monomer instead of the polymer. And what we are doing is that we are adding this nanofiller or nanostructure material in the monomer itself. Okay, and we are adding some initiator to initiate the polymerization reaction. And after adding the initiator, the polymerization takes place. And here, the beauty of this in situ polymerization is that here we will have very good dispersion of the nanofiller into the amorphous structure or into the polymer matrix. Okay, so this in situ polymerization will offer you a very good distribution of this nanostructured material into the polymer matrix. Okay, so I'll just introduce about the polymer. We are very oftenly, we will start our day with the polymers. Okay, so we are very much familiar with the polymeric materials. So polymers are typically utilized in electrical and electronic application as an insulator. We all know the copper wires or wiring, electrical wiring that we are using into our home. It is a copper wire coated with some insulating polymer. Okay, so we are taking the advantage of the insulating properties of the polymer having high electrical resistivity. There are certain typical properties of this polymeric material. They have good strength, flexibility, elasticity, stability, and the most important thing is the moldability, so that we can use them into different forms and shapes. So during my next slides of presentation, I'm going to explain or present the results of polymer nanocomposite. But here we have used conducting polymer to prepare this polymer nanocomposite. So I will very happy to present this slide of the Nobel Prize winners in the chemistry, that is in the year 2000 for the discovery and development of conductive polymer, Professor Alan Heger, Hideki Shirakawa, and Matt Diamond. They got the Nobel Prize for the discovery and the development of conducting polymer. Okay, because we are ever before we used to use this polymer as a well-known insulating material. This is the structure or different examples of conducting polymer, and you don't have to worry about this chemical structure and this, but this is a representative some structure and material. Okay, we can use as a conducting polymer, but mostly I'm going to talk about the polyaniline, that is benzene and alternative nitrogen atoms in between the chain. So we will see conductivity of this semiconducting or conducting polymers. All know that nylon, polyethylene, these are the insulating material. Okay, and they have conductivity into the range of 10 to the power minus 16 to the 10 to the power minus 12 Siemens per centimeter. Okay, silicon, which is a semiconductor, it lacks the conductivity between 10 to the power minus 2 to 10 to the power minus 10. But the conductor material like copper, iron, okay, these are the which are very good conductors. They have the conductivity in the range of 10 to the power 4 to the 10 to the power 6 Siemens per centimeter. While this conducting polymer, they have their conductivity line to alternate their conductivity from insulating region to semiconducting and semiconducting to metallic region. Okay, but changing the dopant concentration, methods of synthesis, so on and so forth. Okay, so those polymers, they have in semiconductor region, but we can tune their property and conductivity in insulator as well as in conductor region, so that conducting polymer we can use as an insulator also, as a semiconducting material as well as conducting material. So these are the certain application of this conducting polymer and its nanocomposite, starting from rechargeable batteries, chemical and particle thermal sensor, polymer, drug release membranes, this control drug release, okay, so on and so forth. So I will give you one small example to illustrate the properties of this polymer nanocomposite by simple changing one component during synthesis process. Here, just see on the screen that I have used paratoluene sulfonic acid to synthesize silver polyaniline nanocomposite. Okay. All other, the components or materials that are same, I have used aniline monomer and aqueous medium and ammonium persulfate as an oxidizing agent. But here I have used paratoluene sulfonic acid as a dopant acid to synthesize this polyaniline from aniline monomer. Okay, so we have characterized this polymer by using UV visual spectroscopy, then FTIR, okay, for the detection of functional group, then XRD to see the phase and the shape and size of the silver nanoparticle into this matrix. Okay, so the right hand side I have presented the efficient images of this PTSA dope silver polyaniline nanocomposite, and here you can see the diameter of this nanorod is approximately 100 nanometer. Okay, so please see the carefully. The structure is a fibrous structure with higher porosity, and the diameter of this nanometer is around 100 nanometer and length up to 2 microns. This we have used by using PTSA as a dopant. But if I change only the dopant from PTSA to DBSA, okay, here all other parameters material are same, except I have selected the dodecyl benzene sulfonic acid, a long or bulky organic acid for the doping of this during synthesis. So here what happened in FESM, you can see that we got the nano fiber material by using DBSA as a acid or as an acidic media, and here the diameter has been dramatically decreased to half of this size as compared to PTSA. Here the diameter of this DBSA to silver polyaniline nanocomposite is around only 50 nanometer. So friends, here you can see by changing only the acid, we can tailor or we can tune the diameter of this nanofibers and nanostructure material to polymer nanocomposite. Okay, for today's application of the silver polyaniline nanocomposite, these are the AB are the same images, while the right hand side AB and CD are the TM images. Here you can see beautifully black colored silver nanoparticles are decorated on the nanofiber of the conducting polymer polyaniline. Okay, so this silver nanostructure is for a polymer matrix we have used successfully for antibacterial activity in collaboration with the university. Okay, and it is observed that the silver polyaniline has shown very good antibacterial and antimicrobial activity for sepsis, microorganism as compared to only or silver nanoparticle. And why this has happened? Because in this thing, silver nanoparticle, okay, the colony grows around that particular silver nanoparticle, while from the silver nanoparticle that we have embedded into the polymer matrix, there is a controlled release of the silver nanoparticle throughout the different time span, and which inhibit and which we have observed very good antimicrobial and antibacterial activity for this nanocomposite as compared to your bulk silver nanoparticle component. In a particular fashion, we have prepared silver nanorod also by using this soft chemistry route, and here you can see very well needle-shaped silver nanoparticles by efficient analysis. Okay, and these silver nanowires or rods we have used for anti-corruption sensing activity. So ever before we have synthesized this silver nanocomposite by bulk polymerization. Now I'm going to introduce how the synthesis process also will going to affect the shape and dimension of the macromolecular structure into nanostructure material. So here I have synthesized polyaniline nanofiber via interfacial polymerization technique. This is in a real sense transformation of a macromolecular structure. We all know the polymer has a macromolecular structure. By using this interfacial polymerization, I have converted this macromolecular structure into nanostructure material. I am sure we all organic chemists are very much aware that organic and aqueous solution or media together, there is a formation of boundary because these two solvents are invisible with each other, right? So here what I have taken the advantage of this properties of the solvents, okay, to prepare the nanostructured polymer. Okay, so here what I have done, I have taken this organic monomer in a media. It is other organic solvent, right? And the ammonium sulfate is oxidizing agent to polymerize this aniline. I have taken an aqueous media to separate bottles. I have prepared these two solutions and suddenly I have mixed all these two solutions together and thereby there is a formation of boundary because there will be a separation of organic layer as well as aqueous. Okay, so the organic layer which contains the monomer molecule aniline come into contact with the aqueous media where there is oxidation and acid present in that media. So here what happened? The front line monomer molecule in contact with aqueous media, right? They will get polymerized, right? And they have the affinity towards water. First front of this monomer molecule which got polymerized, it diffused into the organic media. Thereby, what happens? The second row of the monomer molecule will come in contact with the aqueous media and that will get polymerized. In a similar fashion, successively, all the monomer molecules present in the organic media, they will get polymerized and will have the complete monomer molecule that has been polymerized. So here we can see the boundary between aqueous and organic media, it is in nanometer scale, right? So thereby what happens? The polymer structure, okay, what has to be formed at the interface or boundary has been resulted into a nanostructure material. To our surprise, actually this reaction takes place in a very short period of time, okay? In a few seconds, I can say, or at the most 1 minute. So I have recorded this video and then put into a small, and here you can see the color change, beautiful color change from brown red color to dark green color, which indicates the polymer formation at the interface, right? So after subjecting this nanostructural material for efficient analysis, to our surprise, we observed it has nanofiber having diameter of around 200 nanometer and then up to few microns. Okay, so one of my PhD student who is organic chemist, she has decided that we will use and we will take the advantage of separating funnel, okay, to separate this organic and aqueous media, okay, which we are using our today laboratory experiment for separation of a mixture, and she has put this reaction in a separating funnel, thereby and with precise control of monomer concentration and ratio of rising agent as acid, he has a beautiful control of the morphology and uniform structure of this conducting polymer. So you can see for making the nanostructure material, we don't require any sophisticated technique or instrument or glassware. By using simple laboratory equipment and structures and by taking advantage of chemistry, okay, organic chemistry, we can synthesize nanomaterial. So here I have just presented the transformation of macromolecular polymer structure into a nanostructured material. In a similar fashion, she has looked silver nanoparticle and we have prepared this silver polyaniline coated carbon cloth for different application for antibacterial and antimicrobial application. This is the same image of the carbon cloth that has been coated with silver and polyaniline nanocomposite. In a similar fashion, in situ polymerization, we have prepared cobalt polyaniline nanocomposite. We have characterized it by XR and FTIR, then epicene image, and you can see the morphology, a very beautiful fern leaf-like structure of this cobalt polyaniline nanocomposite, and we have used as an promising room temperature magnetic semiconducting application. Okay, so then now I will proceed with the application of this nanostructure material into a sensor application. So I will go into the application of this nanostructured polymer, particularly conducting polymer, which is a macromolecular structure, but I have converted to a nanostructure or nanomaterial for utility testing application. So here what I have done, I have built an IDT pattern using FRP substrate, that is fiber plastic. Okay, via photolithography technique, and I made this conductive pattern, and on this conductive pattern I have put the drop of solution of this conducting polymer material by simple drop carbon method. Okay, I have exposed this to a different moisture environment or atmosphere, starting from 10% to 100% RH, and it has shown a very very fast and reproducible change in resistance property as we change the environment from low humidity to maximum higher humidity atmosphere. So some of the engineering students, they have tried and attempted for a prototype device by using 9-volt battery as well as LED, and they have presented this device to IIT Technics in the year 2009, and they have bagged the first prize. Okay, so with this nanostructure material, some of the students came pristine or pure polyaniline material, and via FSM you can see the diameter of the nanocomposite has increased or broadened, because here there is a formation of core shell structure. The core part is your carbon nanotube, functionalized carbon nanotube. Okay, and then the other, yeah. The shell part is nothing but your green color conducting polymer. In earlier slide I have shown, here you can see the black color is nothing but your very thin layer coating or envelope material is this polyaniline. Okay, and this core shell arrangement we have confirmed by using the TM images. The dark black color is nothing but the carbon nanotube, functionalized carbon nanotube, and the gray color one or gray color material is nothing but the your polyaniline material. So this highly dispersible and soluble polyaniline carbon nanotube nanocomposite we have used for optical pH sensing application. Here what I have done, this prepared polymer nanocomposite I have just suspended into aqueous media. Okay, and I have ultrasonicated, and I have filled into different sample waters, and I have changed here the pH solution by using pH tablets. Here you can see there is a beautiful change in the color as we change the pH from one that is highly acidic to the highly basic nature, that is pH 12. Okay, so why there is a color change? Because polyaniline is a green color in acidic media because of the emeraldine salt phase of the nature, which is green in color. And we change the pH from acidic to basic. Okay, the structure of the polymer get changed from emeraldine salt to emeraldine base phase, which is blue in color. So by using this carbon nanotube and polyaniline nanocomposite, we can very successfully and easily develop this optical pH sensor. Later we have put this on an optical fiber, and we also have made a flexible one-time use optical pH sensor, just like our pH paper. In a similar manner, we have used the carbon polyaniline coated cotton cloth for wearable sensor application. Here, what we have done in this nanocomposite solution, we have added a cotton cloth, and we have uniformly coated this nanocomposite material onto the cotton cloth, which make the ordinary cotton cloth highly conducting in nature. This is the same image of ordinary or bare cotton cloth before coating the polyaniline nanocomposite by simple dipping and drying process. And after coating, we can see there is a uniform coating of this nanocomposite on the surface of the material. And for the proof of concept or demonstration, we have prepared a molecular motor, okay, by using this cotton thread, and we have connected it to the 9-volt battery and LED to show that we have developed the conducting cotton yarn by coating this nanocomposite material. This is the proof of concept that ordinary cotton cloth is insulating nature, but when we coat it, right, by using this highly conducting carbon nanotube and conducting polymer polyaniline nanocomposite, it will get converted from insulating to highly conducting in nature. And the intensity of the illumination of visibility is a very high and intense unless and until we can disconnect through the 9-volt battery. So as you can see, as we increase the concentration of carbon nanotube from 0.1% to 1%, there is increase in the conductivity of the resulting nanocomposite as well as coated material, and the intensity of the illumination LED is also found in increase, and it is quite natural because here we are increasing the concentration of this carbon nanotube from 0.1 to 1%. Okay, so we are adding this electronically conducting higher or with greater concentration, and thereby the conductivity of nanocomposite is found to be increased. So what is the application? So we can use this conductive microabsorption, static dissipation, resistive and micro-heating, and so forth. And most important application of this material we can use as a sensors. Okay, we can. I have just downloaded some of the applications from the internet. We can use as in the form of a textile-based electrode. Okay, to monitor the ECG, the movement of posture, ECG, electrocardiogram, right? Because at present we are using silver nanogel to have the ECG and brain signals. Okay, so we can use this because for neonatal babies and certain people they have allergy for silver nanogel. So for them we can use this cloth in the form of an electrode or textile-based electrode. So we have published this. So we have filed patent, and media has given a very good publicity for this electronic fabric that we have developed by using nanotechnology. In a similar fashion, we have prepared this conducting polymer-based fiber by using polypyrrole instead of using polyaniline. Here we have changed the conducting polymer to polypyrrole. Okay. Now I'm going to get some other area of the nanostructure material for different application. Okay, so we have developed certain nanostructure material for radiation shielding application. Okay, so we all know when we used to visit orthopedician or a dentist. Okay, then doctor suggests us to have an X-ray of our broken part or broken bone as well as of our tooth. Okay, so the doctors as in the X-ray technician, they as well as the patient, they will get exposed to the X-ray radiation, and there is always a slight risk of damage of cells or tissue or being exposed to any radiation, including low levels of radiation. But the X-ray is very harmful to the tissues for long time exposure. Okay, and they are very harmful, so we should not get exposed to the X-ray radiation for a prolonged time. So at present, this lead containing or lead-based material has been used for X-ray attenuation or X-ray absorption application. But why it has to be removed and replaced? Okay? Because lead, which is being very toxic, and it has great concern from the human and environmental protection standpoint. Okay, lead poisoning is a serious environmental health risk for all humans. Okay, and even if you bury the lead or lead containing material or object, it will contaminate the entire land because of the lead poisoning through the lead containing material. So the lead, mercury, and cadmium, okay, hexavalent chromium, these are the material that has been banned worldwide under the ROHS, that is restriction of hazardous substances. And the most important thing is that why we have to remove the lead or replace, because lead is very costly and it is very, there is it's a heavy material, okay, because it is bulky. So, what is the need for the development of lead-free X-ray absorbing material? So, the most important thing is that elimination of poisonous lead material. The next, second important thing is that reduction in the cost, because lead or lead containing compound are very much costly. And third, the reduction in the weight. Okay, because the doctor community, by wearing this very heavy apron which is made up of lead or lead containing compound, there are chances to develop the bone or backache as well as spine problem for the doctor as well as technicians that use it for X-ray measurements. Okay, so these are the project and objective of this X-ray absorbing material. I will just keep this slide, but I will directly come into the different materials that we have synthesized for X-ray application. We have synthesized barium bismuth sulfide nanostructured material by using hydrothermal technique. Okay, so we have characterized by XRD, then FEC and EM analysis, and here you can see observe the nanostructure material of around 200 to 300 nanometer diameter, nanoparticles, and up to few microns of this barium bismuth sulfide material. For a larger scale synthesis of this material, we have used reaction calorimeter. Here we can have a precise control of pH, temperature, addition of the reactant, this reaction, and we have this reactor at our laboratory. Okay. So we have carried out a larger scale batch of the barium bismuth sulfide by using this reactor, and here also very uniform size this barium bismuth sulfide nanorod having a diameter of around 200 to 300 nanometer size. In a similar fashion, we have synthesized other white colored X-ray absorbing material, which is nothing but the barium plutonium or barium carbonate mixed phase of this material. Okay, this we have synthesized by combustion technique at 60 degrees centigrade, and the left hand side you can see the recipe to prepare this white color X-ray absorbing material. Later on, we have added it into some formulation that we have developed by using SDR plasticizer, and mix this all component using this nanostructural material and uniformly coated on a cotton cloth. Okay, so here I will just show the important the invention that we have made by using nanostructural material. At present, we are using lead containing apron, which is very heavy. The weight of this apron is around 5 to 7 kg. It is just like a wearing silver. In Marathi, we used to call it silver. This is very heavy. Our doctors and X-ray technicians, they are presently use this kind of apron which contain lead containing compounds. While cement has totally replaced lead from this material, we have used some eco-friendly oxide and sulfide material as a material. The formulation by using some their organic vehicle or media, put it on a cotton cloth, and in a similar fashion we also have developed a prototype apron. Here the important and notable thing is that the weight of that apron is just 2.2 kg. Okay, the commercial lead containing toxic apron is around 5 to 7 kg. The apron developed by CMET by using lead or non-lead eco-friendly nanostructure material apron, it is just 2.2 kg. Okay, this is a very useful invention of our doctor and medical community for the use of X-ray attenuation, and we have totally replaced the lead from these materials. Okay, similar we have prepared the curtain also, and these curtains will have application airport for baggage scanning application. Right, we have prepared those also. We have filed patent for this invention, Indian patent, US patent, and PCT also for different countries. And you can see in the picture, the slides, our students' projects are very happily wearing this apron developed by CMET because this is a very light in weight. And at the same time, we have measured the X-ray attenuation at different kVp, and it has shown very good X-ray absorption or attenuation similar to a lead containing apron, and we got it certified from Atomic Energy Regulatory Board, okay, which is the certifying for the X-ray and X-ray containing compound at Bhabha Atomic Research Center, Mumbai. Okay, we got the certification from them, and this prototype apron that we have developed by CMET Pune, it is equivalently X-ray attenuating or absorbing as compared to your commercially lead containing aprons. Okay, in a similar fashion, we have developed glass nanocomposite. Here we have quantum dot of bismuth sulfide. Okay, 2 to 5 nanometer size quantum dots bismuth sulfide nano material into a glass matrix. Okay, my colleagues and our director general, who is an expert in the glass nanocomposite, I am not expert, so I will not comment on this much, but definitely this has a very good application for room for X-ray room for viewing applications. Okay, so exactly when we convert this bulk size particle into nanostructural material, just we are trying to predict it by schematic. Here, the particles are with close packing, but in between some white space. Okay, and when the X-ray pass or get bombarded on the bulk structural material, some of the X-rays will escape through these loopholes or void space in between the molecules. Okay, while in nanostructured material there is a low spectrum of this material or nanostructured object. Thereby, there is a layer oxide, that is lithium cobalt oxide, is a traditional material and cathode material. Anode is nothing but your MCMK or graphite. Electrolyte is nothing but the lithium hexafluorophosphate that has dissolved into electrolyte solvent, that is ethylene carbonate and dimethyl carbonate, that is EC and DFC. Okay, while the white color separator film is nothing but the polypropylene membrane, okay, or polyethylene macroporous membrane to separate this electrode. This is a typical principle of operation of this lithium ion battery, where oxidation and reduction chemical reaction takes place. That means during charging, the lithium ion get intercalated from cathode to anode, and during discharging, it is transferred from anode to cathode, vice versa. Okay. So our parent ministry has funded this project. Under this project, we have proposed to develop a battery. Okay, starting from in-house developed cathode active material, anode active material, with a battery capacity of around 100 to 120 milliampere hour per gram, having the cyclability of 150 to 200 cycles and voltage between 3 to 3.5 volts. Okay, in order to support our defense sector or our military areas, we have proposed to develop a battery that can be operated at -10 to 60 degrees centigrade, so that these lithium ion cells, which we are developed indigenously, starting from its active material, that is cathode and anode, with the entire battery fabrication facility, can be used for military application in Rajasthan Siachen region, where temperature is to fall down up to -10 or even -30 degrees centigrade. Okay, so this is the objective of the project. Okay, so under this project, we have developed lithium cobalt oxide indigenously, starting from the precursor and oxide and so on and so forth. Okay, yeah. This is also, you can see different geometry of the cells, the specification that we have developed by using the facility. Okay, there are lithium-ion battery cells. Okay, this is a roadmap that we have proposed, starting from the inception of the project of the current year, say about 2021. Okay, we also are working on the development of flexible battery. What is flexible battery and why we need the flexible battery? Because nowadays, hello, am I audible? Hello, sir. Sir, I'm audible to all of you. Hello. Hello.
Yes, sir.
Okay. Today the point of electronics research has been
Hello.
electronic device. Hello.
I request all participants mute your mic. Don't disturb the sir. Okay, Milind sir.
Yeah. Okay. Thank you. So, because nowadays there is a trend of flexible electronic devices coming to the batteries. That means, the electronic gadgets like your TV screens and even mobile phone also in future you can see that are bendable or twistable in nature. Okay, and to power these electronic devices or gadget or electronic, materials, we need the energy storage devices like a battery also to be flexible in nature to power this device. These are certain futuristic applications. But nowadays, I can say these are coming into the marketplace also, right? That is flexible LEDs, then imperceptible electronic devices, binding patch on our body skins, okay, RFID tag. Even electronic has developed the bendable and flexible cell phone, that is mobile phone, which has already captured the European market, and very soon it will going to launch in India as well. Okay, then smart electronic, smart watch cells that are also to be visible in nature. Technology in USA, they have already developed this flexible solar cell. So, this electronic gadget and devices, we need batteries also to be flexible in nature. So for that matter, we have wrote a project proposal to MIT, and we have got a sanction for that, and we have developed the flexible battery for this electronics. I mean, different gadgets upcoming futuristic technology application. There is a very good market for this flexible battery, right? So what is the necessity of this all solid state flexible battery? Because currently, or in conventional lithium-ion batteries, the problem is that they limit their scalability, particularly because of safety, lifetime cost, and power density. Okay, and in the present lithium-ion batteries, we are using combustible, electrolyte, that is LiPF6, that has been used along with some organic solvent, okay, and which will lead to its explosion. Okay, so when scientists are trying to develop a very safe, okay, and all solid state batteries that operate at different temperature, very safe. Okay, so in the recent work that has demonstrated that lithium developed by electrostatic nanofibers, that is cathode material, anode material, and separator that we can use by electrostatic technique with excellent overall performance, large capacity, high discharge rate, capacity, and extended cycle life. So by using electrospinning technique, we can prevent highly porous or nanostructure nanofiber material. We already have developed the electrospinning setup at masses. Here you can see we have prepared this okay, this is in our
Namaskar.
Namaskar, namaskar. Very good morning to all. Today we have this second session, and it gives me a great pleasure to introduce Professor Dilip Puri. Sir is one of the most enthusiastic researcher, and more than this, he is a very good teacher. Sir has completed his MS from Garba College and PhD from History Department, University of Pune. Sir has performed postdoctoral work in the University of Bologna, Italy. Sir has a recipient of many fellowships such as a fellow of Indian Academy, fellow of Maharashtra Science Academy. Sir has received the Best Teacher Award from the SPPU, Biggest Book Award from the SPPU. Sir has also received award from the Royal Chemical Society. Sir is the visiting professor of University of Regensburg, Germany, visiting professor of University of Bologna, Italy, and Sir has completed more than 3 major research projects funded by CSR and UGC. In the credit, has more than 170 research papers, and all papers are published in the high impact factor journals. With this brief introduction, I request Professor Double to start his presentation.
Thank you, sir. I will just start sharing my PowerPoint presentation. Just allow me to share it, sir. Visible?
Yeah, it's visible.
Okay, no problem, sir. My voice is clear.
Yes.
Okay, thank you, sir, for kind introduction. Good morning, all of you. So, you had a nice lecture by Dr. Nitin Kulkarni on different information techniques that are used to analyze the applied products, and in fact, CMET is doing really well in the applied research. And now with the 1.5 hours of lecture, we'll talk about the spectroscopic techniques that we use for the field in chemical and the biological sciences. And in this, I would like to focus more on the NMR spectroscopy. This is the basic of the spectroscopy, NMR spectroscopy techniques. You will realize that in the chemical analysis, there are three broad categories almost invariably apply to major areas like fundamental research, product development, product quality control, monitoring, control of pollutants, medical and clinical surveillance. So we are quite aware that this technique, qualitative analysis, just defines identifies one or more species present in a sample. Quantitative analysis where you have a quantification of the sample present in the other components present in the sample. In addition to that, structural analysis that helps to find the three-dimensional spatial arrangements of the atoms in a molecule. At the same time, it also helps to find out the different type of groups present in the molecule, by which you can define the molecule whether it is acid, base, neutral, symbolic, and so on and so on. So all these structural analysis helps in structure determination. In addition to that, you will notice that surface analysis. This is also playing a role in material research, particularly related to physical properties such as topography, depth profiling, orientation of molecules. In the same fashion, in the biological science, you'll notice that microscopy, confocal microscopy also playing a role in order to look at the different type of microorganisms, their structures, shapes, and the different type of biological properties. In a broad way, these techniques, they are divided into these class of compounds. Okay, analysis through spectroscopy, that involves the UV, IR, NMR, and the mass spectrometry. Analysis through chromatography, that involves starting from the thin layer chromatography to preparative thin layer chromatography, gas chromatography coupled with the mass spectrometry, then HPLC, high performance liquid chromatography, and many other flash chromatography. Many different type of instrumentations are available. Okay, actually these chromatography techniques are mainly used to purify the compound to a single component. Okay. Then we have analysis through thermal energy. In this molecular TGA, DTA, and many other instruments related to the thermal energy. Analysis through X-ray technique is also important. For example, single crystal X-ray, powder XRD in the material characterization particularly. Single crystal X-ray is used to find out the three-dimensional structure of a molecule. Then X-ray techniques also involve the field emission scanning electron microscope, then HRTEM or TM itself for the material characterization. Then analysis through microscopy. I think these microscopy techniques are mainly used for the biological purposes. And particularly, I came to know that many of the participants, there is some noise coming from someone. Yes, sir. Sangtaga? To mute the mic. Okay. Then we have analysis through microscopy. Here we particularly look at the biological instruments like a confocal microscope and so on. And yesterday we had a nice visit at the central instrumentation facility that has been created on the Pune University campus. Then analysis through electrochemical techniques and likely chronometry, many electrochemical equipments are there. And in addition to that, there are some miscellaneous techniques are also there. And you will realize that modern advances in these techniques coupled with the tremendous development in computerization, have reached extraordinary levels of sensitivity and sophistication in analysis of the chemical and biological components, and that are important for these two areas. My focus is particularly on the chemical analysis because I am an organic chemist, okay, working in the Department of Chemistry for many years, okay, and being working in the Department of Chemistry, we work in the area of synthetic organic chemistry as well as natural product isolation and so on. But main focus is that look at the natural products and their analysis of this. You will notice that plants, microorganisms, animals, humans, all provide the different type of drugs or the lead structures for the treatment of the wide variety of diseases. Okay. In fact, you will realize that more than 60% of the medicines or the drugs that are at present we are using directly, 10% are directly from the natural products, 30% are the derivatives of the natural products, 20% are the mimics of the natural products. In these mimics, you will notice that we just consider the pharmacophore of the natural product, and nearly 20% are the synthetic. Okay. And here also, synthesizing the molecule, we look into the nature-inspired molecules, and we look into the pharmacophore, and these pharmacophores are identified by the biological scientist. Okay, and in short, you will notice that the natural product is inspiring for all of us, particularly in deciding what type of medicine to be used. In this, I mean, there is a very nice category in the sense that plants, the natural product isolated from the plants are many other terpenes, alkaloids, fatty acids, sugars, I mean, many other compounds, starch, cellulose, everything, not only the medicinal compounds, but all these are derived from the plants. But microorganisms, you know, they are very special category. They are mainly synthesizing antibiotics, okay. And you will notice that including penicillin, first isolated antibiotic penicillin is also isolated from the microorganism. In addition to that, you will also notice that animals are also producing the toxins, and sometimes these toxins serve as a pharmacological tools for our day-to-day life. Human neurotransmitters and hormones are also valuable leads for more potent drugs. And if you look at the nature derived medicines, I would like to tell that all these natural products have very nice three-dimensional chemical structure with a high degree of complexity. Look at this simple example, that's of the Taxol. At present, this paclitaxel is available, being given during the chemo for the person suffering from the cancer, is applicable to all breast cancer, lungs, and different type of cancer diseases. This is a Taxol. This is also isolated from your tree, okay, by cutting full one tree of more than 50 years. We can isolate few milligrams of this Taxol, okay. And then this Taxol at paclitaxel and some derivatives of this Taxol are now being used for the cancer treatment. There is a very nice distinction, particularly compound derived from the marine sources. Look at the complexity in the marine structures. All these are the polyketon type of compound, but look at the
Hello.
hello.
The slide is not moving, sir, please.
I am not moving the slide at present.
Okay, okay.
I mean, these are the polyketides, okay? And of course, if you want to analyze the structure, so we have to purify this compound first by chromatography, and the spectroscopic techniques are used in order to characterize and to solve the complex chemical structure. And in fact, you will notice that not only the complex structures like this one, but complex structures of the biopolymer, proteins, enzymes, big polymers are nicely characterized by using the different techniques. And in this direction, I would like to talk about spectroscopy techniques that are involved. Madam, now I move the slide. Is okay? Yeah. Fine. So I will just talk about the spectroscopic techniques, okay? And you are already aware. This presentation is mainly focused for the teachers who are trained in the biological sciences, okay? So chemistry people may, I mean, chemistry teachers may excuse me because this could be the basic for them, but I would like to consider this for the biology because more than 60% of the teachers are from the biological sciences. So this is the broad spectrum of the spectroscopy, and in the spectroscopy, which is nothing but the interactions of the light with the sample, okay, and this light is nothing but the electromagnetic radiations having the wavelength property. And we start from the cosmic gamma rays. That's the utility of the gamma rays. Okay. For the cancer, as of now, many hospitals have a gamma treatment. Okay. Ultraviolet visual, that it comes with low energy. Infrared, use of infrared tremendous in the day-to-day life, including if you start any device, for example, TV, we use not yet that remote is the infrared. Okay, microwave, and then radio frequency. Here is a very low energy associated with electromagnetic radiations in the radio frequency. And we are looking at the nuclear spin transitions. Okay. So this is a broad spectrum of the electromagnetic radiations. And looking at the spectroscopic techniques that are commonly used in the determinations of the not only the organic but the compound from the plants, microorganisms, and so on. So commonly what we do that we
Hello, sir. 1 minute. Sorry to interrupt, but your slides are not moving. We are still first slide is there.
now it is moving. Yes, sir.
No, not.
Okay, I will just stop it now. We close first. Okay. And again, I will join. This अलीकडे slide आली का?
हा फर्स्ट स्लाइड आहे सर.
Second. आली का second?
नाही एक थोडा वेळ नाही आले अजून तरी नाही आले.
आता आहे?
नाही.
आहे?
अगोदर सर प्रेझेंटेशन करा आणि मग स्लाइड शेअर लागा. म्हणजे अगोदर शो लावा म्हणजे हा प्रॉब्लेम येणार नाही.
हा तसंच मी अगोदर स्लाइड शो ला ओके परत बंद करतो सगळं. सर ओके. Huh?
Yes, no problem.
Yes.
Okay, fine. So, at the spectroscopic techniques that are used for the structure determination, so sample for the investigation is subjected to the radiation, and depending on the source of the light, okay, if you look at the UV visible, then of course we are looking at the pi to pi star transition or the sigma to not sigma, n to pi star transitions, and there this UV visible spectroscopy gives the information about the chromophoric system present in the molecule. What is mean by chromophoric system? That means the presence of carbon-carbon double bond in the conjugation with the another carbon-carbon double bond, or the carbon double bond in conjugation with the carbonyl group, aromatic compounds, heteroaromatic compounds. So these nitriles, okay, C triple bond and nitro functionality, alkyne functionality, carbon-carbon triple bond. In addition to that, we can also look at for the oxochrons that are present on the aromatic compounds. Oxochrons are nothing but the substituents like OH, OMe that increases the lambda max from UV to the visible region. So particularly UV visible gives the information about the chromophoric system present in the molecule. If source of radiation is in the range of IR spectroscopy, then this IR spectroscopy helps to find out the presence of functional groups in the molecule. And you will notice that we can get the information about the presence of OH, then amines. Particularly, we can differentiate the amines like a primary amine, which gives the doublet in the range of 3300 cm-1. Secondary amines gives the single peak in the range of 3300. The tertiary amines, they don't give any signal. You can also find out the functionality like a carbon-carbon double bond, carbon-carbon triple bond, carbon-nitrogen triple bond. In the carbonyl variety of these compounds, starting from the aldehyde to the ketone to the esters to the amide, acid chloride, acid anhydride, acetic anhydride, or the anhydrides. All these are nicely differentiated by looking at the IR spectroscopy. While if the source of radiation is electromagnetic radiations in the radio frequency range, that is nothing but the NMR spectroscopy. Okay, in the NMR spectroscopy, we get the information about the different types of hydrogens. Now, particularly I am making here hydrogen because we are looking at the 1H NMR spectroscopy. But we can also look at the different type of nuclei like carbon 13, N15, O17, and so on in the molecule. We can also look at the relative numbers of the different types of hydrogens, the relative number in the sense that whether hydrogen is attached to carbon is a CH3 type, CH2 or the CH, and that carbon is attached to the oxygen or sp2 carbon or sp in nature, or carbonic carbon, aromatic carbons. So hydrogen attached to the carbon, what type of different type of protons are there, and what is their rating numbers, whether it is a 3, 4, 5 or so. Normally not more than 3. Because carbon has a valency 4. We can also look at the electronic environment for the different types of hydrogens. So this electronic environment is varied depending on the type of electric constituent present in the molecule. In short, we will notice that the NEMA spectroscopy gives the information about the number of the layers. And in this NEMA spectroscopy, we can tentatively assign a structure molecule. In addition to that, recent development, particularly 1960s, 70s onwards, and the H1 NMR spectroscopy was developed in 1945 and then extensively used for the structure determination of the organic molecules. Then 1970 onwards, the era of pulse techniques, where we can also record the NMR spectrum of carbon 13 and 15, F19, fluorine 19, P31, along with the two-dimensional NMR spectroscopy. These two-dimensional and the three-dimensional NMR spectroscopy and the combination of the 1H carbon 13 NMR spectroscopy is now turned out with a powerful tool for the structure determination. But please remember that all these techniques are complementary to each other in the sense that we have to take help of each and every technique. A combination of these three, along with the mass spectrometry, it forms a powerful device in the determination of the structures of organic and the biomolecules. Another important point about this spectroscopy technique is these techniques are non-destructive. Look at the earlier techniques. Most of the techniques, particularly before Second World War, 1950s and so on, 1950, most of the techniques that were used for the structure determination are destructive techniques. In the sense that the compound is getting destroyed during the analysis. But here, record UV and we can recover the sample. Record the IR, we can recover the sample. Record the NMR, we can recover the sample. Okay, and therefore these techniques are called as a and it's non-destructive. And it's very important because cutting full tree of more than 50 years, we are getting few milligrams of the compound. If we destroy that sample during the analysis, then it's of no use. Therefore, this technique has gained lots of importance. Talking about the NMR spectroscopy, you notice that in the early days, the H1 NMR spectroscopy was being used as a technique for the structure determination of the organic compound. Then the pulse techniques have been developed. We can record the carbon 13, N15, O17, and so on. I mean, the nuclei which are having the low natural abundance. We cannot record the C12, we cannot record O16 because those nuclei are non-magnetic in nature. Their spin number is I is equal to 0. But their isotope like carbon 13 and 15 can be recorded. This is the carbon 13 NMR of one of the compound. In the subsequent period, this pulse technique is used in the two-dimensional and the three-dimensional way. This is the representation of the two-dimensional H1 Cauchy spectrum of this compound. Okay. Then subsequently, the three-dimensional pictures. Look at the way that the NMR representation is turning from here to here to here to here. Okay. This is the spectrum of a complex biomolecules, a protein structure, and the credit goes to the Utrecht, who got the Nobel Prize for the structure determination of the biomolecules. The fourth Nobel Prize went to the Paul Letterberg, and the Sir Peter for magnetic resonance imaging. So this is the story, the history, and we must salute them. Why we wanted to salute them? For look at the 1945. I mean, before that, the technique has been developed, okay, for the structure determination, and of course, Bloch and the Purcell, they shared the prize, the first Nobel Prize in 1945. Then the first NMR of that of the ethanol was recorded in 1950s. Then the second Nobel Prize, this is, sorry, one point I wanted to mention here, that look at the period, 1945 they developed the technique, but it is within 7 years of span, the first Nobel Prize came into the NMR spectroscopy in 1952. Both are physicists, please remember. Okay, and world has recognized the importance of this technique within 7 years. Normally to get the Nobel Prize is a contribution of more than 20-25 years. Okay, but world has recognized the importance within short span of the period, and it is found to be true because in the subsequent period the second Nobel Prize went to the Richard Ernst for the discovery in the pulse techniques and so on. Then in 2002 for the Kurt Ulrich, German scientist, for structure determination of the biomolecules, and then the fourth Nobel Prize awarded to the Paul Retrober and the Sir Peter for magnetic resonance imaging. And this is the magnetic resonance imaging. Okay, we get the images of the brain, we get the images of the different tissues and so on. The principle in the NMR spectroscopy, principle in the MRI is one and the same. Please remember, while when this MRI was developed, they were thinking of giving the NMRI nomenclature to this one. But people are afraid for this nuclear word, because Second World War, you know that what happened in Japan. Okay, and therefore they put this nuclear word. Actually, the principle is the same. In the NMR spectroscopy, we are putting the sample here, okay, from in the middle, and you have seen yesterday in the central instrumentation pattern, the sample is loaded and so on. Here our body is inserted in the magnet. This is magnet. And then there is a yes, radio frequency coil is here. Yes, this is the radio frequency coil. Also, radio frequency generator is there. We have seen yesterday radio frequency generator. So what happens that here we look into the H1 carbon-free nuclei. The same H1 nuclei that are present in our body in the form of water. Okay, this water is trapped in a different way, in different ways in the tissues. Okay, the doctors are in a position to diagnose the trapping of the water in the tissues for the normal being who is not suffering from any disease, and the patient who is suffering from disease, and that water trapped in the certain tissues, okay, has certain deformity, and from that image that they are getting, the doctors are in a position to diagnose that patient is suffering from cancer. So this is one technique. Remember that which was applied for the basic research in the earlier, fundamental research, and then subsequently applied to the humans for the identification of the diseases in the body. Okay, so now with this introduction, we'll go to the basics of the NMR. What I will do that we'll just a little go and go a little fast so that you can understand, because you're already aware the NMR spectroscopy. But if you have any difficulty, you can, I mean, raise the point and then we can discuss it. Okay. So what is happening in the NMR spectroscopy is that certain nuclei in the of the molecule, they act as a tiny bar magnet, and when they are placed in a, when they are particularly sample, you take any sample, for example, on the table, then those nuclei, they are randomly oriented, because those ethanol contain the CH3, CH2, and the OH. The H nuclei, they are randomly oriented in all directions. But the moment you place this sample, a strong magnetic field, this is the H0 magnetic field strength, then these nuclei, they get oriented in a two different Okay, low energy and the higher energy excitation energy level. Okay. Populations of the nuclei in the ground state are obviously quite large. However, there are certain populations of nuclei also present in the excited state. According to the Boltzmann distribution, it is just one nuclei among millions that are in the excited state. Okay. Then what happens is that when this nuclei are oriented in this manner, okay, this is the energy required for the, I mean, from the lower and the higher energy levels, okay. Then we supply the electromagnetic radiations in the radio frequency range. When we apply this radio frequency, in that moment, if this energy supplied by the electromagnetic radiations in the radio frequency range, this energy is matched to that of the energy required for the flipping of the nucleus spin from lower to higher energy. At that moment, resonance takes place and nucleus spin undergoes the flipping from lower to higher excited state, and we get this type of flipping of the nucleus. Okay, and in NMR spectroscopy, we are measuring this energy required to flip the nuclei from ground state to the higher energy. This energy in the different nuclei, that means we are considering the 1H nuclei, this 1H energy associated with the different type of protons attached to sp carbon, sp2 carbon, sp3 carbon attached to carbonyl carbon, aromatic carbon is totally different. The difference in the energy of this type of nuclei is very, very minor, the difference. But still we are in a position to take that change in the energy, and we are in a position to get the idea about the electronic environment surrounding the proton. And this is the basic of the NMR spectroscopy. Okay, any doubt in this? And particularly H1 nuclei is having spin I is equal to 1/2, because you know what happened is that this is very simple. Okay, any nuclei, they spin on their own axis, and spinning of this nucleus, it gives two important magnetic properties. One is the spin angular momentum, we call it as a spin quantum number, and the magnetic moment mu. And particularly certain nuclei like the N14, fluorine 35, they also have an electric quadrupole moment. And because of that, the NMR spectrum is very complicated one. That's the reason we cannot record the NMR spectrum of the fluorine 35 and so on. But F19, P31, H1, these they have a spin quantum number, they have a magnetic moment mu. And therefore, when they place in a magnetic field, okay, they precess. And there are two different type of spin states that are available according to this equation. And I mean, there are different type of rules, but I don't want to go into the more detail. You will realize that H1 nucleus has a spin number 1/2. Then we can also record the boron 11, 3/2. We cannot record the carbon 12 because the spin number I is equal to 0, magnetic is equal to 0, and therefore we cannot record the NMR spectrum of the carbon 12. Though it is naturally more abundant, it is 99.8% in the nature, and carbon 13, which is just 1.1%, we are in a position to record because it has a spin number 1/2. N14, though spin number is 1, it has an electric quadrupole moment, therefore we cannot record N15, but we can record the N15 because spin number is 1/2. Oxygen 17, sorry, oxygen 16, which is a naturally abundant. Okay. It is also magnetically inactive. I is equal to 0. Magnetic moment is also 0. Therefore, we cannot record the O16, but we can record O17. We can also record F19 sitting on 29 P31. We are really fortunate that in the organic molecules, the carbon 12 and the O16 is magnetically inactive, because most of the organic molecules molecular formula is containing the carbon, hydrogen, and the nitrogen. Okay, if these elements would have been the magnetic in nature, the H1 NMR spectroscopy would have been no use to the organic to the structure determination of the molecule. And we are really fortunate that carbon 12 and the O16 is 0, I is equal to 0, non-magnetic in nature. So we can record the H1, carbon 13, N15, O17, F19 and so on. Okay, these are the certain properties that are required for the scanning the spectrum. Okay, but forget about this one. Why we cannot record the N14 and the silicon is because it's not spherical in nature. Well, all these nuclei, H1, carbon 13, F19, they are spherical in shape. And therefore, then there is a even charge distribution in this. In this, they have a ellipsoidal prolate or the oblate shape, particularly in case of deuterium, N14, chlorine, and so on. And therefore, in addition to the magnetic moment, they also have electric quadrupole moment. And because of this electric quadrupole moment, the NMR spectrum is very, very broad, not possible to interpret, and therefore we cannot record the NMR. This is the basic NMR equation. These are the energies associated with the different type of states that are available. One is the low energy state and higher energy state, and the energy required for the flipping. It depends on the magnetic mu and the strength of the applied magnetic field. This is the strength of the applied magnetic field and the magnetic moment of the individual nuclei, like H1 has a this magnetic moment mu. Okay, I will come to that point. So this is the delta E, that is the energy required for the flipping of the nuclei from lower to the higher excited state. Is the twice mu is 0. And this energy is supplied by the electromagnetic radiations in the radio frequency range. And therefore, the equation is like this one. Delta E is a Planck's constant equal to twice mu into H0. Therefore, if you do this at the resonance, frequency is equal to twice mu H0 upon H, and this is the basic NMR equation. Frequency, that is frequency or the energy required for the flipping of nucleus, is equal to gamma H0 upon 2 pi. What is this gamma? Gamma is the gyrometric ratio between the magnetic moment mu and the spin number I. Okay, it's called as the gyrometric ratio. This gyrometric ratio is different for the different nuclei. For H1 nuclei, irrespective of the electron moment, it is same, but it is different for carbon 13 nuclei. Okay, carbon 13 nuclei gamma is different. And so, if you know the gamma value, if you know the magnetic field, we can calculate the operating frequency of that instrument. For example, use this equation, and for example, 7 Tesla magnetic field strength. Tesla is the unit that is used to express the magnetic field strength. Actually, Tesla comes little later. First is the Gauss. Gauss is the magnetic field unit. Then kilogauss, and then Tesla. And you must be aware that Tesla is a very important company in the US. And of course, Tesla cars are very famous, very high quality racing cars and so on. Tesla is manufacturing those cars, and they are coming in the market with the electric cars. In fact, already they are there in the European and the American market with Tesla. The same Tesla. Okay. And it is equivalent to the ton, 1 ton in the gram, kilogram, and the tons. So it's a Tesla is the unit for the magnet. So if you have a 7 Tesla magnetic field strength, and if you know the gyrometric ratio value, for example, H1, the gyrometric ratio is this one. Put the values here, then it is a 300 megahertz NMR instrument. Please remember 10 raised to 6. Similarly, we can calculate the operating frequency for carbon 13. If you do so, because carbon 13 now, gamma is 0.6, 1/4 of the H1. Same magnetic field strength, 7 Tesla. And if you put all these things, the value is coming to be 75 megahertz. So please remember, when you record the NMR spectrum, if it is H1 NMR and 300 megahertz NMR instrument, so all NMR instruments are, estimated at their operating frequency. In our department, we have a 300 megahertz NMR instrument, 200 megahertz NMR instrument, 500 megahertz NMR instrument. So we have a 3 NMR instruments. But if you go to National Chemical Laboratory, if you go to ICER, they have a 600 megahertz NMR instrument. Largest NMR instrument in Tata Institute of Fundamental Research, they have a 900 megahertz NMR instrument. okay, because Tata Institute of Fundamental Research is one of the prime institute in India, where the Dharmati, who recorded the first NMR spectrum at the Stanford University, he returned back to India, okay, and joined the Tata Institute of Fundamental Research, and he has developed a big group of NMR spectroscopists who are working in designing and finding out the applications of the NMR spectroscopy. Okay, so all NMR instruments are named after the operating frequency of the H1 nuclei. But please remember that the operating frequency for the other nuclei in the same NMR instrument is different. If it is a 300 megahertz NMR instrument, the magnetic field strength is 7 Tesla. In this 7 Tesla, operating frequency for carbon 13 is a 75 megahertz. Okay, so this is what exactly I wanted to mention here. 500 megahertz NMR. Yesterday you have seen this 500 megahertz NMR. You have also seen the 400 megahertz NMR instrument. But look at this 500 megahertz NMR instrument. 11 Tesla magnetic field strength. 100 is the operating frequency for H1. You can calculate the operating frequency for carbon 13 as 125 megahertz. F19 it is 470. It all depend on this barometric ratio. And for N15, the operating frequency is 50 megahertz. So please remember, when you write in the and by publishing the papers, don't get confused. Write properly the operating frequency for H1 and the operating frequency for carbon 13. Approximately, the carbon 13 operating frequency is 1/4 to that of the H1 nuclei. If it is a 500, 1/4 of the 500 is a 125. 1/4 of the 300 is a 75. 1/4 of the 100 megahertz is a 25. Nowadays, these NMR instruments are not available. Now, I think the Oxford has come up with the permanent magnet, the tabletop model for the graduate and the postgraduate students. It's costing around 30 lakh rupees and so on. Just permanent magnet and tabletop model, and they are available. But earlier days we used to use it because in our department we were having the 90 megahertz NMR instrument, 100 megahertz NMR instrument. But nowadays nobody makes use of this NMR instrument for the research purpose. Only 300 and above instruments are available. And it is all operating frequency depends on this barometric ratio, which varies from one to other. And normally in the CSR, these type of questions are being asked. But let's forget about that. We already seen this one, continuous wave NMR, Fourier transform also you have seen. In the Fourier transform NMR spectroscopy, the sequence that is used is the pulse acquisition and pulse delay. Mr. Kharade, you explained to you about the continuous wave NMR machines and the Fourier transform NMR machines. So I will skip this one. This is what exactly the 40 megahertz NMR spectrum with the electromagnet and so on. This is the electromagnet. Okay, this is the 1960s continuous wave NMR machine. This is the superconducting magnet. Okay, here 100 megahertz NMR facility at the Savitribai Phule Pune University. Please remember that this facility is generated from the support of the Savitribai Phule Pune University funding. And you will to know that in those days when I was the head of the department, management council of the department of the university, this proposal was submitted to the Senate as well as the management committee. The management committee trusted upon me. They are given more than 25 crore rupees to establish the center instrumentation facility. And I'm really happy that this facility has came up in a very nice way. You already visited online, but you are most welcome to visit on offline. Okay. Many students in those days, particularly, we started working, I think, in 2013. Okay. But 2013 to 2019, I mean, before pandemic, many students of the BSc, MSc, they used to visit our department and visit this center. So you are also most welcome to visit the center instrumentation facility, which is pushing more than many different type of instruments. Look at the NMR spectrum that has been recorded in those days, and record the NMR spectrum was recorded here. Okay, any difficulty? Yes. Any participants have any
Yes,
difficulty?
biological science peoples, particularly
Yeah,
chemistry peoples.
tell me
Yeah.
what is the difficulty?
Yes, sir. You can proceed.
Acha. Okay, now with this, we are getting the NMR spectrum. Okay, normally we submit the 5 milligram of the sample. Please remember, don't submit more than 5 milligram or 10 milligram of the sample uh, to the central instrumentation facility in case of 1H NMR. For carbon 13, yes, 50 milligram, 100 milligram is okay. But weigh the sample properly. Otherwise, there is a relaxation processes that are there. Because of the relaxation process, the sample concentration is very high, then NMR spectrum is very, very broad. So don't submit more sample, weigh the sample on the balance, and don't submit more than 10 milligram of the sample for the 1H NMR. When you submit the samples, you get the NMR spectrum like this one. Okay. Actually, this is a very old NMR that I have put it here because now no one gives this integrated. There are this NMR spectrum is interpreted by the three different ways. And if you look at the NMR, okay, then you will notice that the number of signals is 1, 2, and the 3 types of signals indicate that there are 3 different types of protons in the molecule. And you will notice that this is a simple example of the ethyl methyl ketone. Okay, you will notice that these CH3s are different, these CH2s are different, and this CH3 is different. So you can differentiate. Look at this one. This CH3 attached to the carbon is different. This CH3 attached to this carbon is different. So number of signals indicate how many different types of protons are there. The location of the signal, where it is here, it is here, it is here. It indicates the electronic environment surrounding the proton. What is the electronic environment here surrounding the proton? The intensity of the signal, look at this one. This is little smaller in intensity. This is very tall in intensity. But this intensity indicates the number of protons of that type responsible for the signal. And you will notice that this is a 1 line, but this is a 3-line pattern, this is a 4-line pattern. So this splitting of the signal indicates the protons on the adjacent carbon atom. For example, this CH3 has another carbon adjacent to it and has a 2 protons. So by looking at this one, you know that there is a spin-spin interactions, and we can make out the number of protons on the adjacent carbon atom. In short, we can detect the neighbors and the neighbors of a functional group. So what is neighbor to this? What is neighbor to this? What is the neighbor to this? Okay. So we can find out this type of feature. And the NMR spectrum is analyzed based on this red line is the integration line. It gives the information about the number of protons responsible for the signal. Nowadays, we'll not get this type of spectrum. You get the FID, pre-induction decay. That pre-induction decay, you have to analyze at your place using the spin top software. Okay. If it is not there with you, then we'll support. We'll give. But normally, what we do that we send this FID by mail to all the persons who are looking for the NMR spectrum, all the teachers, researchers, and so on. And in that case, the integration is given here in the form of 1, 2, 3, 4. Then this is the integration. The second is the chemical shift. Chemical shift is the position, okay, where these are coming. And the coupling constant is the split. These are the 3 parameters based on which we can interpret the NMR spectrum. magnet, okay, and they got this spectrum. But how they confirm that this signal is corresponding to CH3, this is CH2 and this is OH? Can you imagine? Tell me, this is a story in 1950s. Anybody can guess how
Sir,
they confirm that? Yes.
sir, your PPT is not visible now.
I mean, PPT is not visible. Is okay? Sir? Hello? Hello, sir?
Just a minute, sir. Some- No, no, your PPT is not appearing.
Now?
No, sir.
This is strange, why it is happening like this one. I have done nothing. Okay, we'll just cancel it again and restart it. I never faced this type of problem. The first time I'm facing this problem. Is okay, sir?
No, sir, till it is not visible.
Is okay?
Yeah, it is appearing. First slide is there.
Yeah. Is moving?
Yes, yes, it is moving.
Okay. Yeah, I was asking a question, this. So, okay, sir? Yes, sir?
Yes, it is looking.
I mean, this is the NMR spectrum that has been recorded in 1952 by Arnold Dharmati and the Packard. Dharmati was working with this Packard, the Arnold, at the Stanford University. He came back and established the powerful NMR division at the Tata Institute of Fundamental Research. Tell me how they came to the conclusion that this signal is corresponding to the methyl, this corresponding to the CH2, and this corresponding to OH. At present, we are in a position to know by looking at the height of this signal. Okay. But in those days, can you imagine how they must have done it? Yes, it's a question to all the teachers.
No hydrogen molecules, sir.
No, but how we can decide they got this spectrum? How to decide which is CS3? What technique they must have used it? Huh? Weak area. What they did, they have taken the paper and they cut this portion from here to here, here to here, and here to here. And they made these papers on the microbalance. What they noticed that if this paper is suppose 0.01 milligram, so this is a 0.01 milligram, this portion is 0.01 milligram, and this portion is 0.03 milligram. And from that they confirm that this is the CS3, 3 proton, this is the 2 proton, and this is the 1 proton that is OH. And they turn out to be correct because subsequent integration is also showing the same. So this is the way that the integration helps to find out the number of protons responsible for the signal. And nowadays we get this height. So what you can do, once the spectrum is there, the height of the signal is given here. Okay, now they give it like this one, the bottom of the spectrum. And for example, this is the signal. 3 plus 2, 5 plus 3, 8 total number of protons. Now you calculate the integration. 4 plus 6 plus 6, total integration is 16 mm. So for 8 hydrogen, the integration is 16 mm. So 1 hydrogen is corresponding to 2 mm. So there are 4 mm integration. Therefore, this is a 2 proton signal. It is a 6 mm integration. It is a 6 protons, 3 protons signal. This is also 6 mm integration. Therefore, this is also 3 proton integration. Okay. So this is the ethyl methyl ketone. This CH2 is 1 2 proton. This methyl is this one, this methyl is this one. So we can just, this is a normal way of doing that one. Please remember that we get the ratio of protons. Normally in the integration, the ratio of protons, relative ratio. And then we can elaborate this ratio of protons to the actual number of protons. For example, look at the T-butyl and methyl ether. Okay, this is the T-butyl mixture. We get the two singlets. Now in this case, there is a one single corresponding to two spaces, one single corresponding to 6. So ratio is 2:6. 1:3 ratio. But actual number of protons responsible are 3 and the 9. So look at molecular formula and the ratio of protons, we can convert this ratio of protons to the number of protons. Is clear to you or not? Yes. So integration gives the ratio of protons present in the molecule. That ratio of protons you can subsequently elaborate by looking at the molecular formula of the compound. Here the ratio, integration ratio is 1:3, but actual number of protons is 3:2. So this integration helps to find out, number of protons responsible for the signal, but it also helps to find the mixture of compounds present in the molecule. For example, if the solvent contains chrome and the ethanol, yes, we can identify what is the mixture of compound. In addition to that, this integration helps to find out the enantiomers in the case of parallel catalyst and the diastereomers that we are getting in the different reaction sequence. Organic chemists are aware of the diastereomeric ratio, so we can very easily find out the diastereomeric ratio. We can also find out the ketoenol tautomerism in the molecule. So this is the integration. Okay, now we turn our attention to the chemical state. Actually, chemical state, why these protons they result at the different position? Actually, if you look at the equation is like that: frequency is equal to gamma h0 upon 2. Now in this, frequency is constant, gamma is also constant, h0 is also constant. If this is the scenario, then all the protons should resonate at one and the same place, and NMR will be of no use to the organic chemist. But this is not the scenario. The scenario is something different. The scenario is like that. If you look at the nuclei, any nuclei you consider the H1 nuclei. For H1 nuclei, there is one electron surrounding this nucleus. This electron, which is present and on the periphery of the nucleus, when it is placed in a strong magnetic field, it creates an electric current. And this or electronic current, then subsequently, of course, it moves perpendicular to the applied magnetic field. And then this creates an induced magnetic field, which is in opposite direction to the applied magnetic field. And therefore, this induced magnetic field protects this nuclei from the applied magnetic field. And this electronic environment, particularly hydrogen, is covalently bonded to the thing. Okay. And this electronic environment surrounding the proton is different. And therefore, different type of induced magnetic fields that are created. And as a result, different protons are resonating at the different positions. Therefore, this equation is rewritten as a frequency equal to gamma 0 upon 2 pi into bracket 1 upon sigma, where this sigma is nothing but the shielding constant. To what extent this nucleus is shielded because of the electronic environment? This is a shielding. Okay, shielding, protection of the nucleus by the electron. This sigma is the shielding constant. Okay, and this value depends on the local electronic environment. More the electron density in the proton, more is the shielding, more the protected proton, and therefore it will result in high field site. And less electron density is the de-shielded is the proton. The same example, if you go to Himalaya, okay, where the temperature is -10 degrees centigrade, but your body not feeling exactly -10 degrees centigrade. Why? This is because your body is protected with the sweater, muffler, overcoat, and so on. So that is shielding. Okay. More electron density, more is the shielding. That's what exactly the shielding is. So that's the reason different protons they are resonating at the different positions. Look at this one. Now simple ethanol again. Okay. So look at this hydrogen. Why these three hydrogens are different? Now this hydrogen is directly attached to oxygen. Oxygen is more electronegative. Okay. Therefore, it will attract the electron towards oxygen. As a result, the electron density surrounding the proton is less. Therefore, it is appearing at the low field region. Look at these two hydrogens. Hydrogens are attached to the carbon, and in turn, this carbon is attached to the another oxygen heteroatom. Now, oxygen will withdraw the electrons from this bond. In turn, will withdraw the electrons from this bond, CH bond. Therefore, this CH will experience there is a less electron density. Therefore, they are coming here. While these three CH3s, they are one carbon away as compared to the electron withdrawing oxygen. And in the chemistry, the inductive effect, electron withdrawing inductive effect is practically negligible when the proton is one carbon away. And therefore, these three protons, they are not experiencing any little effect is there, no doubt, but very, very less. And therefore, these protons, they are resonating here. So that's the reason different protons, they resonate at the different positions. The is not the absolute position. In UV and the IR, please remember that we are getting the absolute lambda max. In the UV, in the IR, we are getting the absolute value. Carbonyl frequency, for example, which is ester carbonyl coming at 1740 cm minus 1. That's absolute. But in NMR spectroscopy, there is no proton without any electron. Therefore, we are not getting the absolute position. And therefore, in order to overcome this difficulty, you will realize that this relative position of the protons is measured with respect to some reference standard, which is normally placed at 0 of a charge. And with respect to this 0 of a charge, you will realize that the positions of these signals are made. And in that case, spectrometric science is used in most of the NMR spectrum. Please remember, they use the DTO as a solvent. In that case, tetramethylcyan is not soluble in the DTO. Marker standard. So sodium salt of some acid compounds, but pertaining to the silicon. But if you are using the methanol, acetone, benzene, then you can make use of the tetramethylcyan as a compound. There are many advantages of this tetramethylcyan. The most important is that this is a volatile. Okay, the point is 27 degrees centigrade. Therefore, we can pour the sample just removing the tetramethylcyan. And secondly, the important point is that this has a unique line position where normal protons and organic are normal molecules. Okay. But some silyl derivatives in the organic chemistry. There they are interfering with the TMS, no doubt about this. But otherwise, most of the organic compounds, proton signals are resonating away from TMS. Okay. And therefore, has a unique position you can explain by looking at the electropositive nature of the silicon, which pushes electron to the carbon. Carbon is more electronegative as compared to silicon. It attracts the electron towards it and it pushes the electron to the hydrogen. All these hydrogens are highly shielded. They are coming on that highly shielded position. Remember, now both of the organic molecules they resonated between, for example, in the most of the compound different protons will resonate between 0 to 1000 Hz. If the 300 megas in a most of the organic compounds between 0 to 3000 Hz. If the 500 megas in a this is a high frequency of this is the lower frequency. Remember, so this is the third is the unit that is used to express the location of the signals in the NMR spectrum. Okay. But the major difficulty associated with this chemical shift expressed in hertz is that this hertz value is instrument. Okay, look at this one. 300 megahertz. The hertz value is very. 100 megahertz hertz value is different. So hertz are hertz value is the applied magnetic field, the operating frequency, and therefore being there are a number of instruments, can calculate this frequency one instrument, other instrument using this equation. Okay. But in order to avoid this calculating business, okay, another use another scale that is used is the data is equal to shift in frequency from k divided by in hertz. Please remember, megahertz is 10 raised to 6 hertz. Okay, and therefore, in order to avoid point not multiplied by 6, that is this delta value is called as the parts per million. So, and remember, we are measuring one part in millions, and that is the reason why the NMR instruments are very, very expensive. The important point of this is the chemical shift expressed in the value is dimensionless, is not proportional to the operating frequency. 60 megahertz NMR instrument is 60 hertz, but in 100 megahertz NMR instrument, this CS2 ethanol will resonate at 100, but delta value will remain the same. That's what exactly the delta value irrespective of the okay, we can calculate the delta value. Here the value chemicals lower the delta is the D shielded photon. Lower the value of chemical shift hertz of the data. Shielded photon. These are the different type of organic molecules. They are present in the different region. Aliphatic, alicyclic. That means full CH3, CH2, CH2, CH2. They will resonate here. Then beta substituted aliphatic. That means beta substituted means alpha and the beta. It will come alpine. They are coming between 2.5 to 3 delta. Carbon triple bond. Protons attached to carbon-carbon triple bond. Monosubstituted like a ethanol OH attached to CH2. They are coming between 2 to 4 delta. If they are disubstituted compound, then they are more decelerated. Carbon double bond protons are coming from 4.5 to 7.5. Aromatic and the heteroaromatic, they are coming from 6 to 9 delta. And the aldehydic proton is the one which is most decelerated. OH proton normally, please remember, is coming anywhere between 0 to 15 delta. OH, hydrogen attached to the heteroatom like oxygen, nitrogen, sulfur, and so on. We must know the equals of the protons. Look at the acetone. This CH3 and this CH3 is the same, therefore is one equivalent. Therefore, we'll get a one signal. Dimethyl here, one signal. Here also one signal. Okay. But if you compare all the position of this methyl, for example, 2.4 delta, this will be around 3.3, and this will be around 3.6 delta. We can easily differentiate the position depending on the location of the signal. We can very easily differentiate whether it is acetone or methyl ether or methyl carboxylate. So these are equivalent. But if, for example, look at this ethyl chloride. These CH2 protons are different from this CH2 because CH2, these hydrogens are the electron differential. Therefore, two signals. Here also two signals. These two are identical. These three are these two signals. Particularly in the aromatic, you will notice that depending on the substituents here, the most substituted benzene, there are three signals. These we call as the ortho position. They are same meta position, para position is different. There are 1, 2, 3 different. If it is disubstituted, these two are same. If the disubstituted but identical, in the X and Y, the situation is different. Or disubstituted two signals. Ortho meta disubstituted three signals. This is one type, second type, and third type. Para disubstituted. Okay, both substituents are identical and therefore all protons are identical. So by just looking at the number of signals, we can make out very easily whether it is orthodisubstituted, metadisubstituted, or paradisubstituted, provided both the substitutes are identical. If the substitutes are different, then the situation is different. Also, but both X and Y, then there are 4 signals here. There are so metadisubstituted 4 signals. In case of paradisubstituted, where both the substituents are different, 2 signals. Same thing you can get in the. There are different parameters that affect the chemical shift. One, the electron withdrawing effect, hybridization, von Roll's effect, diamond shielding, and the resonance effect. But please remember, the actual chemical shift value is the result of a combination of these effects. Okay. The actual value is the combination of these effects. It's not only one effect. But for that you must know the other effects. Just simply I want to mention that CS attached to some R, but here R is equal to carbon. Okay, sp carbon 0.9. CS2, which is having the both the C carbons on the both the sides, 1.4. It is deshielded. Why it is deshielded? Because carbon is more electronegative as compared to hydrogen. Here we are replacing one hydrogen with the carbon. Okay. Now the carbon is more electronegative as compared to hydrogen. Therefore, deshielding 0.4. Same thing here, deshielding. Same analogy is applicable to all of them. CS3 attached to carbon 0.9. CS attached to the nitrogen 2.4. Because nitrogen is electronegative as compared to the carbon. It withdraws the electron to it, and hydrogens are deshielded. Oxygen is more electronegative as compared to nitrogen. Therefore, OCS3 comes at 3. And CH3 attached to nitrogen 2.4. But CH attached to the nitrogen 2.8. And CH attached to the nitrogen, CH as a 2 substituent 3.2. Same scenario case of oxygen also 3.3 here, 3.6, and 4 delta. Around 0.4 difference, 0.4 delta difference is there. Carbonyl group is also different from that of oxygen. Now CS has to carbonyl 2.1. So we can very easily differentiate acetone from that of the methanol. Methanol, methyl come at 3.3, acetone, methyl 2.1. Look at the CH2 attached to the carbonic 2.5 and CH attached to the carbonic 2.9. So electron withdrawing effect is one of the factors that affect the chemical shift. Stronger the electron withdrawing group, more is the density. This all depends on the electronegativity of this substituent. This is what the electronegativity. Oxygen electronegativity is 3.5 as compared to nitrogen, which is 3. Carbon is also electronegative 2.3 as compared to the hydrogen 2.1. Okay. Please remember this scenario. Same scenario is applicable to halogens also. Look at this different period. And for example, CSCS is low form 7.3 delta. Okay. So this is what exactly the different protons at different positions. OH proton there is OH, NH, and the SS proton they appear between any position between 0 to 15 delta. Okay. And this position is dependent on variety of these parameters like the substrate, concentration, temperature, and body. Okay. But good trick to identify OH protons or the NH protons is that add a drop of DTO. Add a drop of DTO. Shift the NMR tube and rescan the NMR spectrum. What happens is that this OH is getting exchanged with the DTO. It gets converted to the ROD. Therefore, that signal will vanish. OH proton will disappear. And if there is a simple proton, that will get there is OH related of the NH proton. And we get a HBO, one signal is coming at 4.6. So that's a best trick to identify the OH NH. But you know, this OH signal has a position anywhere between the 0 to 15 delta, and many times that interferes. If you change the solvent, if you change the concentration, the OH position will vary. And that's what exactly. In the chloroform, CDCl3, it's coming at 2.5. But in DMSO, it is coming at 4.3. The variety of factors are there that affect the chemical state. These are the normal solvent, deuterated acetic acid, deuterated acetone, deuterated acetonitrile, benzene, deuterated chloroform. We cannot use the undetected solvent because otherwise that CHCl3 is used. The itself will give the big peak at 7.25 where all the protons will get worse.
ಎಷ್ಟು category असेल काय बिल
Hello, sir. Hello, sir. Yeah. Hello.
हा आवाज येतोय सर, खोटेकर सरांनी म्यूट केला.
Okay, okay. There are different type of solvent that are used. Please remember, for example, CDCl3 is the usual solvent, but CDCl3 that we are using is not a 100% deuterium incorporation. Okay, this deuterium incorporation is 99.9%. So whatever the 0.% the CH is present, that will give the signal at 7.5. Please remember, by looking at this position also, we can make out this data value. There's a signal at these data values, we can make out which solvent is used in our carry. This is what exactly the why we are getting the signal. Actually, we should not get this signal. It is 100% deuterium incorporation. 100% deuterium incorporation. CDCl3 is very nearly 3 times costly as compared to 99.9%. 99.99% CDCl3 is available. That is 3 times costly. CDCl3 5 ml is required to it is costing 100 rupees. Please remember. Chloroform 0.5 ml is just less than rupee. But CDCl3 0.5 ml is 100 rupees. That's the reason we are charging 150 rupees to record the NMR spectrum. This is just to, I mean, that is the amount that is used for solvent to record the NMR spectrum. So even though 150 rupees, you feel that it is very heavy amount. Of course, it is 150 rupees, good amount for carrying one person. But we can't do anything because 100 rupees just wasted in recording the NMR. We are not asking anything else because operator is there, instrument, and so on. These are the solvers. Another factor is the aminotropic seeding and the deseeding effect. This is particularly created when the double bond report then aromatic ring is present in the molecule. This is the result of the electronic circulation of these 5 bonds here. Here also there are 5 bonds. And the circulation of these electrons, they create induced magnetic field. Okay. The proton is situated where the induced magnetic field is parallel to that of the applied magnetic field, then that proton is shielded. Therefore, olefinic protons are shielded. In case of acidic protons, the orientation of acidic protons is in this fashion. This is because circulation of the electrons has to be perpendicular to the applied magnetic field. Circulation of the electrons, which is perpendicular to the applied magnetic field. And therefore, look at this hydrogen situated where the induced magnetic field is opposite direction to that of the H2O. Therefore, acyclic hydrogens are shielded as compared to the olefinic hydrogens. Aromatic hydrogens, there are many create strong ring current, and because of that, these protons which are situated in the close vicinity of this circulation of electron or the induced magnetic field, they are in the parallel position to that of the applied magnetic field, and therefore they are heavily shielded, coming at 7.25. And then we can also make out the position. For example, this proton is coming in the shielding zone of this double bond. This proton is shielded compared to this proton. These are isomeric compounds, okay? But we can differentiate by looking at the NMR spectrum. A number of cases like this one. Okay, look at this methyl in this terpenoid. 0.74 because it's coming in the shielding zone of the carbonyl group. There are n number of examples like this one. The diamanity anisotropic effect is also applicable to the carbon-carbon double bond. Look at this hydrogen and this hydrogen. This and this hydrogen will have a different positions. Now, third factor is the resonance effect, and this you can very nicely explain by looking at the simple example of the olefin compound and the substituted olefin compound. If it is electron donor, then this proton has a deshielded because of the electron withdrawing in the triplet. So 5 point this proton is equal to this proton. 5.7 is coming to be 6.38. While this proton, which are coming at high delta, now there is an electron donation. Therefore, there is a net negative charge that is possible at the beta carbon. It is shielding, therefore 3.85. While in electron withdrawing substituent, beta carbon as well as alpha carbon, they are deshielded, 5.85 and 6.4, like this. This is the resonance effect that because of the hydrogen are shielded and deshielded. I do not want to go more details, but same situation in the case of aromatic compounds. Depending on the substituents, the ortho, meta, and the para protons are played and the deshielded. So we can differentiate these chemical shift and the positions of the protons with idea about the electronic environment surrounding them. Third parameter that is there is a spin-spin coupling. Okay. Now look at this example, ethyl methyl ketone again. Now you notice that this CH3 is coming as a single line, but this CH3 is coming as a 3-line pattern. Why this splitting is there? And this splitting is, this is the elaborated spectrum. The first and the second line, and second and the third line is called as the coupling constant J, and this J is always expressed in terms of Hertz. And this Hertz, the coupling constant J expressed in Hertz is not proportional to the climatic field. This is the J value. And corresponding CH2, if you look at it, this is the CH2 4-line pattern. This is the J value here. Please remember all these multiplates are symmetric to the midpoint, which is corresponding to the chemical system. Here also we find the same J value 7 Hertz, here also 7 Hertz. So this is a result of the spin interaction between this CH3 and the CH2. This CH2 will experience the different spin orientation of this CH3. As a result, this will have a different magnetic experience by the protons. Similarly, this CH3 will experience the different magnetic field strength because of this different spin orientation of this CH2. This is called as the spin-spin interaction. Spin-spin interaction resulted into the splitting of the signals. Okay. This is what the. But please remember the equivalent proton, they do not. There are 3 rules based on which. Okay, there are 3 different types of coupling constants. One is the geminal coupling, vicinal coupling, 1, 3, 1, 4, and the 1, 5, and the 1, 6. So there are 3 rules based on which we can identify the number and the nature of protons. And the number and the nature of signals. For example, why there is 4 lines here for this CH2? Why there is a 3 lines for this CH3? There is a single line for this one. It is governed by these 3 rules. One rule is that equivalent protons do not activate with each other. These 3 protons here, they do not couple with each other because they have a same electronic environment. Therefore, they do not couple. These 2 protons, they do not couple with each other. These 3 protons, they do not couple with each other. But these 3 protons have a carbon attached to it, have 2 hydrogens. Both they will interact with each other. And when this means interaction result in multiple lines. And the maximum number of lines given by the equation 2 NaI plus 1, so on, where NaI, Na, and B and C are the number of neighboring non-equivalent nuclei, and I is equal to their spin number. In case of H1, F and the P3 nuclei, I is equal to 1 half. Therefore, maximum number of lines can calculate by equation Na plus 1 to Nb plus 1. Okay. That means for this CS3, there are two neighbors, therefore 2 plus 1, therefore 3 lines. For this CS2, there are neighbors, therefore 3 plus 1, 4 lines. And the intensities of the lines are calculated based on the coefficient of expression x plus raised to 1. That means if there is one proton of adjacent carbon atom, then the equation becomes 1x plus 1. So coefficient are 1:1. That means doublet will come with the intensity of 1:1. If there are two neighbors, this methyl there are two neighbors. Okay. The equation becomes x squared plus 2x plus 1, and therefore the equation coefficients are 1:2:1. Therefore, intensities of the lines is 1:1. That's you will notice based on that you can calculate the number of lines in the NMR spectrum. Since how much time I have?
You can
Hello?
get till for 5 to 7 minutes. Up to 12, 50 will complete.
This is the way that we can find out the number of signals in the NMR spectrum, whether it is stimulate, doublet, or thing. Different values are there for the coupling constant. Example, this CH, no neighbor, this CH has a one neighbor, lines. This CH has a two neighbors, this CH has a three neighbors. And you notice that the intensity also goes, but the two is the same. If it is one proton integration, for these two lines integration remains the same. Single line integration remains the same. So this is the way that spin coupling constant helps to find out the neighbors and the neighbors. A number 3, but I will stop at this point. And just take an example. The diagram is being characterized. In this case, I would like to do example of curcumin. The main ingredient of this curcumin is this compound called curcumin. And now we isolate from this curcuma longa. This is the botanical. We isolate from this one. After isolation, we record the UV is the range of 350 to around something, okay, depending on the solvent that is used. It indicates that this compound is having the unsaturation, that means carbon-carbon bond in conjugation with the carbon group. Then we record the IR. IR is the carbon-carbon double value, aromatic carbon-carbon double value, give the value of the carbonyl groups and so on. So from that, I can find out the different type of OH. This is the OH. So we can find out the percentage that is present. Then we record the NMR. This is the NMR. By looking at the NMR spectrum, these are all olefinic protons as well as the aromatic protons and the PTOC. This OH is coming anywhere between these two, but we can make it, we can find it out by looking at the deviation. Okay. So looking at the splitting pattern, we can decide where this OH is, this there, and so on and so on. We can also record the carbon 13 NMR spectrum. This is the carbon 13. Look at the value 0 to 20. Here for proton 0 to 10. The carbon NMR is there. And in the carbon 13, then is the NMR. But it is a single one dimensional. And also the CH to CH. Then we have the mass spectroscopy. We can also record the mass spectrum and the spectrum and not spectroscopy is spectrometry. Okay, and from the mass we can make out the molecule of the compound and the fragment that we are getting. We also find out different type of fragments how we can join together to get the structure. So these are different techniques UV, IR, NMR that are being used with the mass spectrometry to identify the compound. Same thing we collaboration with IBB. Of course, uh, there are number from our group as well as from IBB doing collaboration, isolation of the compounds from the plants as well as from the uh, microorganisms, and we identify the structure of those compounds by using this. So these are the techniques that are routinely used to identify the compounds in the chemical as well as the biological science. With this, I will stop at this point and go to the synthesis of the question and answer.
Okay, sir has done very nice presentation on NMR spectroscopy. Now I declare that the session is open for the discussion. If anybody has any query, you can ask to the sir. Okay, in the chat box there is no any question. Hey, anybody have question? Participants? Hello. Am I audible?
Yes.
Yeah. I think there are no questions. If you have any questions, you can mail to the sir. Okay. So Megakar is asking question. Please explain principle of FTNMR.
I think Kharade sir has explained it, but I can also explain to a certain extent. In the continuous wave NMR machines, that means the NMR machines of the old generations. Okay. Those NMR machines, what we used to do that we used to observe one frequency at one particular time.
Yeah.
In the same that we used to record the spectrum from one end to the other end. Okay, and in that you will notice that we are observing one frequency at one particular time. Okay. Yeah. Look at this one. Vary this either frequency or magnetic field strength. Okay. And by recording the NMR spectrum, okay, here you will notice that the output device of this NMR is on the x-axis, and we vary the either frequency or the magnetic field. As the frequency varies, using this RF transmitter, the response of the signal is displayed. So we used to record, for example, we start from here. Slowly, we vary the frequency. And then we used to get the signal. Then again, very slowly go ahead. Whenever there is a resonance, we get the signal. Whenever there is a signal, resonance, we used to get the signal. So in the continuous wave NMR machines, we used to observe one frequency at one particular moment. And this recording the one NMR spectrum used to take 10 minutes roughly. Okay. I was operator in those days. I'm talking about the 1980s story. I appointed as a microanalyst and to operate the 90 megahertz NMR instrument. And that time we used to use this plotter, and to plot, and we used to vary the frequency, and by varying the frequency, one particular resonance signal is observed at one particular time. Okay, and this plotting requires a large time, 10 minutes and so on. So this was the scan, but it used to take a long time. In the recent techniques, the Fourier transform technique, what is done is that entire range of the signal of, for example, 0 to 10 delta, we apply a strong, powerful wave frequency pulse. That is the pulse. It's called as the frequency. So rather than scanning one particular frequency at one particular time, 0 to 10, there is a pulse is generated of high width, steep width. Okay, and then because of that, all the protons, different type of protons, they are excited simultaneously at one and the same time. So all the excited protons, time domain phenomena, okay, they are acquired in the computer. And there is a pulse delay, because this is pulse delay is why? Is because the nuclei has to come down to the ground state. And then we apply another pulse. All these sequence it requires just 1 second. Now what happens is that giving one pulse, all the protons are excited and they are acquired or stored in the computer. For example, in case of H1 NMR spectroscopy, we give only 4 or 5 pulses. Okay, that is enough, because H1 nuclei has a very high natural abundance of 99.9%. So that is enough. And therefore, all this pulse and acquired data is there in the form of Flinders-Lake decay. This is called as FID. This is like this one, which is a time domain phenomenon. Now, the Fourier, who is the mathematician, what he did that he has did the mathematical calculations where the Flinders decay data can be transformed into the frequency domain phenomena. Okay, so these absorbed signals are now recorded as the frequency domain phenomena in this fashion. So Fourier transform is nothing but the transformation of one type of data into another type of data. Flinders decay is a time domain phenomena, while NMR spectrum that we are recording is the frequency domain phenomena. So using this Fourier transform technique, we are converting this Flinders decay that is stored in the computer to frequency domain phenomena. And this process takes few milliseconds, please remember. So 10 minutes required to scan the NMR spectrum are now reduced to 1 second. And this is a good technique to record the NMR spectrum of the low abundant nuclei like a carbon-13. Carbon-13 has a 1.1% abundance. And that's the reason we have to do the long pulses of 1,000, 2,000, 3,000 pulse sequence. Give the pulse sequence, one pulse sequence, acquire the data, and the again pulse delay, the pulse sequence. And during this, what happens is that whenever there is a resonance, they are getting added on. And there are 1,000 pulses, all are getting added on, and those are later on Fourier transformed to get the frequency domain phenomena of the spectrum. So this is the Fourier transformation technique, and this is now routinely used in record the routinely used to record the H1, R13, P31, and so on. Is it clear?
One more question is, sir, that biological and chemical compounds, do they have same characterization? Nilima
How we
Devi has
can
asked this question.
see the biological compounds and the chemical compounds. Actually, the biological compounds are also chemical compounds. Okay. Thing is that in the natural product, if you take one tree, there are hundreds of different types of chemicals. In the microorganisms also, there are 100 different types of chemical compounds. We have to isolate, we have to purify the individual component, and then we have to record the spectrum. And the spectrum will be different for the different compounds. So whether it is derived from the biological sources or the plant sources, different chemical compounds will have a different, but it's chemical compound, it is chemistry. It will be from the microorganisms or so on, marine organisms. They are giving the different type of compound nature is the same in the chemical compound. And chemical compounds will have different compounds have a different structure. There is no differentiation like a chemical, biological. Only thing is the source of material will be different. That's all.
Sir,
Yeah.
what is the important modification that is done in FDNMR instrument than in the conventional NMR instrument?
Madam, there are many things associated with the Fourier transform technique. Okay. One thing is that we need the powerful radio frequency oscillator, one thing. Then secondly, we need very high powerful computational technique. Means computer, because we have to acquire this data. Okay, when there is a splitting, this FID will be different type of FID. So we require the data that can be stored, and for this purpose we require very high quality computer, and that's one of the reason why the Fourier transform machines are very expensive. 500 megas in MR instrument, even 300 megas in MR instrument is costing around 1 crore rupees, 1.2 crores. So, of course, there are many other things also, and just like a black and white TV and the color TV, why we are not using the black and white TV? Black and white TV, we just show only two colors, but go for the color TV, we get different type of colors. Then high definition TV. Why we want HD TV? You know, so this type of resolution of the color, resolution of the dots is very important, and that's the reason the FTA machines are very expensive. There are lots of modifications.
Okay, I think questions are over. So
Sir, one question, sir.
yeah.
Sir, when we take this carbon-13 NMR, why we do not get the proton signal?
Yeah, because we use a proton-ion decoupling technique. There is a decoupling technique is there. Okay. So, and please remember that the resonance frequency of the carbon-13 nuclei is different that of the resonance frequency of the H1 nuclei. And therefore, carbon-13 nuclei resonate at the different position. And that's the reason we are not getting the H1. Just like in the TV, we have 100 channels. How we can get the different channels? GTV, Sony different, India Today different, CNN different. Because the operating frequency of the carbon-13 nuclei is different than the operating frequency of H1. But you will notice that carbon-13 also covers the proton. Therefore, carbon-13 that has been recorded in those days is very complicated one. In order to make carbon-13 NMR spectrum more simplified, all the carbon-13 signals are states. This is because we are using the technique called as the proton-ion decoupling technique. Thereby, we are exciting all the protons, and the coupling constant between the C13 and the hydrogen is totally removed, and all the carbons are resolved extremely. And therefore, in carbon-13 recording, H1 nuclei will not come. The frequency is different. For carbon, gamma is different, magnetic moment is different.
Thank you so much.
Okay, so
Any other
it
question?
is now 1 o'clock. So all the questions are answered by sir very well. So now I will take this opportunity. Has explained the NMR spectroscopy, use of NMR spectroscopy in the identification of different types of proton, then coupling, decoupling constant, and the number of signals that are appearing in the NMR spectrum and how to interpret this. From the side of Professor Ramakrishna More College, HRDC, University of Pune, and all the participants, we are very thankful to the student giving such a nice lecture. Thank you, sir. Thank you.
Thank you, sir. Thank you very much.
Rasale, sir, announcement.
I will do, sir.
Yeah, sir. Thank you, sir.
Hello, sir.
Thank you, Rasai, sir. Thank you very much, all of you.
Thank you, sir.
Thank you, sir. Thank you.
I will leave, sir.
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
So there is a break of lunch up to 1:30, and we will start our last session at 1:30 p.m. So take lunch. Hello. Bhavin sir. Okay, good afternoon, all of you.
Afternoon, sir.
Good afternoon. So now we are going towards the end of our faculty development program. So good afternoon, all of you. It's the last session that we have to attend. So on behalf of Professor Ramkrishna Murray College, Pune District Education Association, I welcome today's speaker for last session, Ankita Ashok Nagarkar. Madam has completed their BE Computer and MBA Operation and HR, and at present she is working in Amazon Corporate Office, Pune. She has so many achievements. The first achievement, that is, she has registered 10 patents. She won National Ignite Award for Innovation from 4,156 participants all over Maharashtra. She is won that prize, that is the Ignite Award, at the hand of Dr. APJ Abdul Kalam in 11th November 2011, Ahmedabad. She filed first patent of improvement stapler at the age of 16. So innovation is in his blood. Her innovation got recognized in the Limca Book Record in 2014. She facilitated by so many renowned persons, that is the mayor of PCMC, director of IIT Bombay, Honorable Sharad Pawar, Anna Hazare, Chief Minister Devendra Fadnavis, and so many. She received Paryavaran Sakhi Award in 2019. She is a runner-up in Miss Sakhi Saundaryavati Pune 19. She received Miss India 2019 title in beauty. Also received Maharashtrashi Saundaryavati in 2019 for best hairstyle. So I would like to say she is very small. The Moti Lahan Pan Kirti Mahan. She owned 72 awards. I'm just briefly introduce here. She awarded 72 awards she received. She delivered about 600 lectures. She delivered lecture in Ignite Award function at IIM Ahmedabad, then for ISAR company, then in COEP Pune. Then she is the chief guest for inaugural address in various science exhibition as an youth icon. She gave training to school students in biogas, terrace garden, biomass, solar cooker, solar water heater, solar electricity, and so on. She is the given interviews on telecast, that is innovation on ZTV, interview on innovation on Z24 TAS, G24 TAS, and many other news channels. She also gave interview on at Pune Akashvani on the occasion of National Science Day. She is deputed as a leader in Nehru Yuva Kendra for Youth Development by Government of India. She deputed as Secretary of Pune for National Youth Council of India. She elected as a minister in that is a Young Inspiratory Network of Sakal. So many awards. So I welcome Ankita Nagarkar, such a versatile, dynamic, and beautiful personality in this last session of Faculty Development Program. She is going to talk on patent plagiarism and about research ethics. So I welcome again Ankita Nagarkar on behalf of her college and Pune District Education Association. So I hand over charge to the Ankita Nagarkar. Thank you.
Thank you, sir. Am I audible?
Yes.
So, yes, I am Ankita Nagarkar. And first of all, I would like to thank the organizers and the PD Association for organizing my session and giving me the opportunity to share my views. So, as you all have just heard a brief introduction about me, about all of my achievements, I assure you that today's session will be a definitely motivating session for all of you. So, here the agenda of today's session will be I'm going to share my own experience about how I received these awards, how I filed patents on my name, and how my journey began in the field of research. At the same time, I'll tell a brief introduction about patent patent filing, and further moving ahead will go towards plagiarism and research ethics. So, yes, I would like to start today's session with a small story. So, this story is about myself. I was in standard 10 when I faced a problem. I mean, when I was in standard 10 at the age of 16, I was stapling some school assignment papers. And while stapling the school assignment papers, I faced a problem. So the problem was, while stapling these papers, the pins suddenly got over. And as they suddenly got over, it created a lot of disturbance in my work and a lot of irritation was caused. So I just kept on thinking for a few days that if these pens are, that it would be really better if I would understand beforehand these pins are going to get over. Right? So this was a simple, small problem which I faced, that sudden finishing of the pins. So after thinking for a few days, a concept struck my mind. The concept was, I colored the last few pins of the stapler. So while stapling, when the non-colored pins get over and the colored pins become visible to us, we'll come to know that the pins are going to finish and now you have to insert a new set. So I hope this concept is clear to all of you. This is a really small concept, but I can say it is a really useful one as well. So with this concept, my journey in the field of innovation, my journey in the field of research began at the age of 16. So this concept struck my mind, but after it struck my mind, I did not keep it with myself. I was aware about an institute named National Innovation Foundation, NIF. You can search it on Google as well for further information regarding this institute. So this is an institute which gives exposure to such innovative concepts at a national level. And I was aware about this, and I just thought that whatever concept has struck my mind, I can apply it for the national award, and maybe I can get felicitated, or maybe I can receive this national award. But at the same time, I had a lot of questions running through my mind that whatever concept I had, it was really a small concept, and will it be recognized at a national level? Can I file a patent of this small innovation? I had such a number of questions which I had in my mind. But at the same time, I had confidence in myself as well. So I just thought that I can at least apply it. Either I will get a national award, or else I'll get a learning from it. So this was the approach which I kept. And further, I drafted my innovation in a particular format. There's a specific format for drafting patent. So I drafted my innovation in that particular format. And after that, I applied it to this institute for this national award. And to my surprise, I can say 11/11/11 was the day when I received this National Ignite Award at the hands of Dr. APJ Abdul Kalam sir. So yes, I can say it was a dream come true moment for me, and yes, a turning point of my life as well. Because Kalam sir is my role model, and I had always dreamt of meeting him, of getting a chance of interacting with him, and that was the day when when I got this opportunity. So I would also like to share a small experience which happened there during this award ceremony. So there were almost 4,156 entries who participated in this particular national award, national competition. So out of those entries, only 22 candidates were selected, and out of those 22 candidates, I was the only candidate from Maharashtra who got selected for this national award. So I can say again, it was a proud moment for me because I was representing Maharashtra over there. So this ceremony took place in Ahmedabad IIM, Indian Institute of Management. So the day arrived, and I was there in Ahmedabad in IIM for this award ceremony. And no doubt, I was excited for receiving this national award from my role model, But at the same time, I was a bit nervous as well, because I had, you can say, many questions running through my mind. But then, when Kalam sir visited my stall, I briefed him my innovation. I told the entire story. I told my project, what my innovation was, which I just told you all right now regarding the stapler. So after I told him my concept, he was really surprised. He appreciated me. He gave me a few suggestions, and also he told me a small example. So this example was of an apple. He said me that each and every one of us is aware that apple falls down from the tree, right? It doesn't go up or it doesn't go in any other direction, but it falls down from the tree, right? But no one thought upon it that why does it fall down only and why doesn't it go in any other direction? But it was Newton who thought upon it and came up with the gravitational force concept. Similarly, the stapler thing which I just told you, that problem each and every one of us faces. Even he had faced this problem, but then no one thought upon it that how it can be overcome. And it was I who thought upon it and came up with this solution. So I think as those words somewhere proved to be a motivation for me. And from that point of time, I understood that no innovation is small or big. No research is small or big. If it is innovative, if it is really useful, if it has novelty in it, definitely you can call it as an innovation. So from that point of time, I decided to keep an innovative approach in whatever things I do, right? So, for example, you can say in day-to-day life, we carry out n number of activities. Activities like we go to school, we go to college, we do our household day-to-day activities, right? So, while doing these activities, we many a times face a lot of problems. But what usually happens is that once we face a problem, we try to find out a temporary solution, or maybe we just leave it there and then forget it after a few days. This is what usually happens. But this should not be done. One should always keep an approach which approach that whatever problems we face, we should not leave it there and then. Instead, we should think upon it, and after thinking, we should come up with some innovative solution. And I think that solution can definitely be called as an innovation, right? So that is the policy. I think that is a pathway which I personally follow. Whatever problems I face in my day-to-day activities, day-to-day life, I do not leave it. Instead, I think upon it and try to come up with some sort of solution. Not necessary that solution might be innovative. Not necessary that that solution might not be existing earlier in the market, right? But at least one should develop that capacity, that thinking power, that whatever problems we face, we should try to find out some solution out of it, right? So once this habit gets developed, definitely you can come up with great more innovations. So this is the thing which I personally follow, and therefore after my first patent, I have done 9 different innovations, and at present I have 10 different patents registered on my name. So yes, I would just like to tell in short about my 10 different innovations. So the second innovation is of an innovative petrol pump. So normally, when you go on the petrol pumps to fill petrol, what is the problem which we usually face? Many a times it happens that you do not get that amount of petrol which you have asked for, right? So बऱ्याच वेळेला आपण म्हणतो पेट्रोलची चोरी होते. So similar thing happened with me. I had gone to the petrol pump to fill petrol, but then I noticed that I did not get the amount of quantity of petrol which I had asked for. And so yes, I just thought that this is not right and something should be done upon it. Therefore, I have developed a setup The advantage of this setup is that quality and quantity of petrol is visible, and it can be set up on the existing petrol pumps itself. So what I have done is I have added a transparent unbreakable glass vessel on the existing petrol pump. So normally when the petrol from your tank goes directly into your vehicle, so then the quality and quantity of petrol is not visible, right? So what I have done is I have fit this transparent unbreakable glass vessel in between, so that the petrol from the tank will first go into the vessel, and after the customer sees it, after the customer gets satisfied, then it will go into your vehicle. So by doing this, the quality and quantity of petrol is visible. So I have filed a patent of this, and not only that, but I have also applied this concept, this innovation to the PM office. They have approved it as well, and they have formed a committee of a few petrol pumps. We are dealing with them, and hopefully you will soon see these sort of petrol pumps in the market. The third innovation is, I can say, useful innovation for ladies, for girls who are working in the kitchen. So the third innovation is eye irritation free onion cutting device. As the name suggests, that this innovation is related to something related to onion. So normally when we cut onion, what is the major problem which we face? A lot of tears come from our eyes, right? A lot of irritation is caused. So once I was doing the same thing and I faced this problem, but I did not sit there crying. What I did was I thought that how can this problem be avoided? How can we overcome this problem? And after thinking, I developed a small machine. And by using that machine, if you cut the onion, then whatever gases come from the onion, they will not come in direct contact with your eyes, and ultimately, the gas which comes out from the onion will not come in direct contact with your eyes, and ultimately it will reduce the irritation which is caused. So this was my third innovation, my third patent. The next fourth innovation is I can say I have developed a vehicle which is electricity free and pollution free. So this vehicle is used from coming down from higher level to lower level, and it completely runs upon gravitational force. The next fifth innovation is a mobile phone charger which is wind operated. So normally when you travel long distances, uh, it happens that your phone battery gets discharged. And I can say nowadays, life without phone, life without mobile is unimaginable, right? So what I have done is I have developed a mobile phone charger, but this runs upon wind energy and not electricity. What you have to do is you have to just hold this charger outside the window of the vehicle which you are traveling, maybe a train, maybe a car, or a bus. So as the wind as the wind efficiency, as per the wind efficiency, the mobile phone will get charged as this charger. These blades are connected to your mobile phone using a motor. So more the wind energy, more fast your phone will be charged. So this was my fifth innovation. The next is dish antenna, improved dish antenna. So even now, if you go in various rural areas, you'll observe that all TV channels are not clearly visible. So to make them clearly visible, I have modified the existing dish antenna in such a way that it can be adjusted as per the frequency, and whatever TV channels are not clearly visible, they can be seen clearly. No doubt there are more technical details in it, but main aim of doing this innovation was to make TV channels more clearly visible, even in rural areas. So the next innovation was copper coated aluminium vessels. So if you are aware that cooking or boiling in aluminium vessels is not good for our health. So when I came to know about this, I thought that something should be done as it is not good for our health. Therefore, I've developed copper coated aluminium vessels. The next is herbal liquid for washing utensils. So normally the liquid or the soap which we use for washing the utensils, many a times it happens that it is as it is there on the vessels and you consume it. You eat food in it, you cook in it, and it gets consumed. So again, it is not good for our health. Therefore, I've made a, a herbal liquid which you can say it is an edible liquid. Even if it gets consumed, there is no harm in it. So this was my next innovation. After that, I have developed a multi-dimensional exerciser. So nowadays, if you see, people are like fitness freak, gyms and all are in a boom. So what I have done is I have developed a multi-dimensional exerciser at my home. So this exerciser is nothing but a cycle, but it is not a normal cycle. This, if you perform exercises on this cycle, as it is a cycle, normal exercise is done, but at the same time, your brain concentration power also increases. So this is the advantage of doing exercise on this cycle. It will have a display in front, and on that display, you will see around 17 to 18 forms of exercises. And by seeing those exercises, you will have to perform it on this cycle. And your once you perform it, your brain concentration power slowly increases. So this is the advantage of this exerciser. The 10th innovation is innovative car indicators. So normally indicators, when you see, they are situated below near the headlight. So normally what happens is if this practically implemented it on my own car. So these were my 10 different innovations, which I just told you in short because of the time constraint. But after hearing to my concepts, my innovations, one thing must be clear to all of you, that whatever innovations I have done, they are useful in my day-to-day activity, right? And also they are useful for the society. So one should always keep approach that whenever you're doing innovation, whenever you're doing any sort of research, you should always think in such a way that whatever your innovation is, it must be useful for you as well, but it must be useful for the society also. Right? This is the mechanism of that easy drawing system. So earlier, when the patent was filed for this particular system, the patent was that you pull the rope, all the rods come down, you put the cloth on one single rod, you leave the rope and it goes up, right? So after listening to this, you must have understood one disadvantage in this particular patent. That disadvantage is that even if you want to use only a single rod, unnecessarily all three rods come down, right? So this was a drawback of that particular innovation. But as this drawback was noticed, further advancement was done, and another patent was filed, which was you pull the rope. If you want to use a single rod, only one rod will come down. You put the clothes on it, you leave the rope, and the rod will go up, right? So I hope the modification or the advancement is clear here, right? So in this particular way, even if the similar patent is already existing in the market, you can try to find out some advancement in it, try to do some modifications in it, and definitely you can go for patent filing of that particular innovation, right? So further moving ahead, what is the importance of patent filing? So you all are hearing like from 15-20 minutes about patent. You have heard my own patents. You have heard what a patent actually is. Now I'll just tell you why one should go for patent filing process. What is the actual importance of it? So if you file patent of your innovation, you become the owner of your innovation, right? So ownership is gained. Now, what is meant by ownership? So, whatever concept you have in your mind, whatever innovation you have done, no one can use it, no one can commercialize it, no one can bring it into the market without your permission, right? So, I think this is the power of patent. Even if it is a small concept, the person will have to take your permission before using it, before commercializing it. Now, the stapler thing which I told you, it is really a simple concept. But as I have filed patent of it, as my patent is granted, no one can use it without my permission. And if anybody tries to do so, I can sue the person. Again, it goes into the legal matter, like filing a case. There's some amount of fine which is imposed on the person. So these are the legal matters which come into scene, right? So you are the owner of your innovation. The second point I can say is there is no age barrier. I filed my first patent at the age of 16. You can file the first patent now as well, right? So there is no such age barrier for filing the patent. The next point I can say is there is no educational qualification barrier as well. For example, if you are from medical field, you can file a patent related to electrical field. You can file a patent related to mechanical field. So there is no such field barrier, right? The only requirement is that you must be completely clear about your innovation and it must be valid. It must be legit, right? So there is no such educational qualification barrier as well. Also, I can say your profile becomes strong. So what I personally experienced is that I'll tell you from my own experience that after I completed my computer engineering, we apply for interviews, right, for job. Similarly, I had applied for interviews for getting placements. And what I experienced there was I need not speak much because my CV spoke a lot. Automatically, your profile, your biodata becomes strong. That by just seeing your resume, the point of view of the interviewer changes, and automatically it can become an advantage for you all. Also, I can say you come in contact with all intellectually strong people. So yes, again, it is a great advantage for you all, and it will again give a direction, I can say, for development in this particular field. So yes, your network becomes strong, and the major advantage I can say is that it is a great source of income. So all you need is to have one innovative concept in your mind, and you can gain a great income out of it. Now, how you can gain income out of it? So after you file the patent, there are three ways of dealing with the patent. First way is you can sell your patent. So there are some companies which approach you after your patent gets published. So you can sell your innovation, you can sell your patent to them, and they'll give you some specific amount, right? So after you sell your patent, they are the owner of that innovation. This is the first way. The second way is you can take royalty. Royalty in the sense it is nothing but the partnership, right? So the company might give you like 3%, 4%, 10%, 20% royalty. It completely depends upon your innovation. So second way is you can take royalty. Third way is you can start your own business. So this is what I am going to do of my own patents. I'm going to start my own business. I got a lot of offers as well for buying my patent, but I did not sell my patents. But I'm going to start my own company, and hopefully soon I'll enter into the market being a businesswoman. So this is the third way in which you can deal with the patent. First is you can sell. Second is you can take royalty, and third you can start your own business. So these are the three ways in which you can gain a lot of income just by having one innovative concept in your mind, right? So I can say these are just a few importances which I have shortlisted and why one should go for patent filing. Now, the next point is how to file a patent. So I can say there's an advantage of patent filing. The advantage is that model is not necessary. So at present, if you just have an idea, if you just have a innovative concept in your mind and you do not have model of it, it's completely fine. You can still go ahead and file the patent of that particular innovation, right? So model is not necessary. This is the advantage. Now, if you do not present the model, then what do you present? So you present the draft. So there's a particular format in which you need to write your innovation. So I'll just tell the format in short. No doubt there are more legal words, more technical words which are to be used in the draft. But just in short, for a beginner, I will tell you about what a draft actually should look like. So the draft consists of the following points. The first point is the title. So even if you consider a research paper, if you consider a book, there's a title for it, right? Similarly, the patent should have a title. Now, the title should be in such a way that one should have a clear understanding about what a patent actually is, about what your innovation actually is, right? So, first point is title. Second point is field of invention. So, field in the sense you have to specify how, in which field your innovation is, whether it is mechanical, whether it is electrical, whether it is medical, whether it is computer or what, right? So, you have to specify the field of your invention. The third point is, I can say it is prior art. Prior art is, I can say, very important part of your patent draft. Prior art is nothing but you have to specify how your innovation is superior than the existing innovations. So this thing you have to mention in the prior art. So just to elaborate it more, I'll give you my own example. That stapler pens were already existing in the market, right? But in the prior art, I specified how my pens are useful than the existing pens, how my pens are superior than the existing pens. So this thing I mentioned in the prior art. The next point is working. Working in the sense how what actually your innovation works, how it works, what it actually is. So that you have to specify there. The next point is drawings. So drawings are the most I can say compulsory part. It is a must. You have to specify your innovation diagrammatically. The next is summary. Summary is like as a book has a summary at the back. Similarly, you have to summarize your innovation. And the last point is claims. So claims are nothing but the advantages of your innovation. Again, it is a most important part of your draft. The last point. So yes, this is the I've just told you in short about how a draft should actually look like. Uh, just as a beginner, you can draft your innovation in this particular format, and definitely if anybody has drafted this innovation, they need further assistance, be happy to help them. So yes, this was about the patent drafting. And after that, nothing is the next step is this documentation, right? There are certain forms to be filled, then you can just register your patent. So also one most important point is that you should carry out the search. Search is the most important part. Why? Because as I just mentioned, that patent cannot be filed of the existing innovations, right? So for that, you have to search whether your concept, whether your innovation is already existing in the market or no, right? For that, the search is to be carried out. Now, this search can be the paid search as well as the unpaid search. So there are various sites on Google also you can search it. There are various other paid sites as well on which you can do this patent search. Now, if you consider research paper, uh, there you have to check the uniqueness of your research by using plagiarism, right? Uh, plagiarism is nothing but the uniqueness. But here in the patent, there's no such plagiarism used. all you have to do is you have to carry out maximum possible search on internet and check if your innovation is already existing, if your innovation is already patented or no, right? So this is the process which I just told you in short about uh, how to go ahead with patent filing process, right? So I hope patent filing process is clear to you all. And if not, I can definitely say that uh, if any sort of help is needed regarding patent filing, regarding innovation, I'll be happy to help you all. Because I can say I have delivered more than 600 lectures in various schools, colleges, even multinational companies regarding innovation and patent. And yes, I feel happy in saying that from my own college, there were more than 130 patents which the students have filed on their name. And yes, they are also earning a great income out of it. So yes, I just do it as a social service, I can say, because whatever knowledge I have, whatever achievement I have achieved, I just want to share it with all of you, all the people around here, with the students, with the faculty. So this is the main aim behind delivering lectures, just as a social service. And at the same time, I can say that I feel proud in saying that there's a book written on me named माझ्या अंकिताचा शोध, in which you will get to read my journey in this field of innovation and research. And at the same time, you'll get a brief introduction about what a patent actually is. And yes, this book is written by my mother herself, Sunita Nagarkar. And I feel happy in saying that more than 20,000 copies of this book have been sold. And the third edition is in the market. So, again, the main aim behind writing this book was that to make people aware about innovation and about patent, right? So, therefore, we just gave this book at no profit, no loss base, 50 rupees. And it is, I can say, really a useful book for students, faculties, parents as well. So yes, this was about me, my innovations, which I just try to tell you all in short. I hope my journey was useful for you all as well, and it must have given a pathway about how one should go ahead in the field of research and innovation. Furthermore, to tell about me, apart from innovation, apart from research, I'm also more into social work. I've been carrying out various social activities through various forums. Uh, uh, like through Government of India, I've been selected as a taluka level officer, and like many forums like Young Inspirators Network, National Youth Council of India. So these are the forums in which I work socially. So yes, this was a brief introduction, brief journey I can say about all of my activities. And yes, once again, I would like to thank all of you for giving me this opportunity to share my views, share my experience in front of you all. Now I just keep this session open for question answers. If anybody has any, we can go ahead with the question answer session.
Okay, thank you, Ankita. Now this session is open for discussion. Yes, anybody have any question?
Yes, sir. I have one. First,
Yes, ask please.
very inspiring story, Ankita. I really enjoyed it. I would like to ask if I have found a patent and I want, and somebody else copied my idea after that, then how will I profit from that?
Yes, Ankita.
I'm sorry, I was on mute. So as I said, that there's a legal matter which comes into scene. So it is a crime, I can say, that if anybody is copying your consent, if anybody is copying your patent, because it is like it literally works on the basis of seconds, not even hours, not even minutes. It works on seconds. Like if I have filed a patent, like say 30 minutes and 20 seconds. And if another person files the patent on the 21st, at the 21st second, but still you are the owner of that patent, right? The one who has filed the 20th second. So it was in such a minute format, minute time. So if anybody claims that this is my innovation, and if, or rather you can say if anybody copies your innovation, so you will have to hire a lawyer, and you can file a case on that particular person. And if it is a legal matter, right? So with the guidance of the lawyer, you can file a case, and there's a jail, there's some amount of fine which is imposed. So yes, how will you come to know that the concept is copied? So if the person copies it, definitely will bring it into the market. And once it is into the market, it is open, and there are various ways, maybe through media exposure, maybe through newspaper, you can come to know that your patent is already into the market. So yes, you can hire a lawyer and go into this legal matter.
Hello, Ankita.
Yes.
There is one more question from Megakad. How we would come to know that our patented innovation is getting used anywhere? What are the ways to find it?
Yes, so as I just mentioned, that there are various ways. So if any person copies your innovation, definitely will not keep it to himself, because there is no advantage for him in that way, right? So he will definitely bring it into the market. Once it is into the market, people make an advertisement of their product. They give newspaper exposures. You can see it on television. You can see it in newspapers, various banners maybe, or mouth publicity, right? So there are various ways of publicity which uh, from which you can come to know that your concept is already into the market, someone is already using it and making a profit out of it. So if you come across this situation, as I said, you can go into the legal matter and sue the person.
Other question is, your patent from India or universal patent?
So my patents are Indian patents, but there are two different types of patents. First is Indian patent, and then the second is foreign. Foreign in the sense, there are various countries in which you can file patents. So as we are Indians, we first have to file Indian patent compulsorily. And after that, if you wish to do production of your product in foreign countries as well, you can go for foreign patents as well, right? So my patents are Indian patents as of now.
प्रश्न विचारायचा तुम्हाला प्रश्न विचारायचा मला. Hello.
Yes.
One more question is there. How long the right of patents are protected?
Yes. So the patents are protected. Patent rights are protected for 20 years. So after 20 years, it becomes open. You have to renew it, right? Always. So as per the Indian patent law, the primary rights are for 20 years.
Okay. Can you please suggest some paid and non-paid websites to check already registered patents?
Yes. So the paid sites, all of us do not have sites format while searching on Google. You should not mention your entire concept in detail on the Google search bar, because again the data gets saved and there are chances of it being hacked and it being copied, right? So for example, if you have done an innovation related to a pen, so you can just type on Google pen related patents, and you'll get all the list of patents, the list of all the patents, right? That way you can do the primary search.
Okay, one more question is there.
Sir,
Yes.
may I ask?
Yes, yes.
This is Dr. Padmini Pidiyar, Mahatma Phule College, Pimpri. Ma'am, I just want to, is there any app for the protection of our patent that which can give us whether it is hacking or used by another person?
No, actually, there's no such app here as of now, but maybe in future may come into scene.
Okay, thank you, ma'am.
There is one more question, madam. What does it mean to license a patent and why it is done?
I'm sorry, can you please repeat?
What does it mean to license a patent and why it is done?
Okay, so
License a patent.
yeah, so licensing is nothing. Actually, licensing a patent is to gain the rights of your patent. So once you file the patent, once you register it on your name, you get a patent number. And once you get that patent number, you can say that you are an owner of that innovation, right? So you need not wait for the patent to be granted. So granting of the patent, it takes some time. It is not an immediate process. After you file a patent, it goes into publishing stage, it goes into examination stage. After that, the decision is given if it is granted or rejected, right? So you need not wait till then. After you register the patent, you get a patent number, and you can call it as a license, right? So it is really important because until and unless you get that, you cannot say that the patent is yours. So that is a major part I can say.
How much expense required for patent file?
So expense, as in I cannot tell you a specific amount because it completely depends upon innovation. It varies innovation to innovation because there are n number of factors which are included. It may include the type of the patent, whether it is complete or provisional, or maybe the number of claims, number of pages which you have included, and so there are such various factors that are included. But to give a brief, just a range I can give you, it may be around 25,000 to 40,000. But again, as I said, there's no such specific amount. So it depends upon various factors, like various forms are to be filled. Every form has a different amount. So yes, so the expense changes. It varies.
How many days required to grant a patent?
So it is not actually days, it is actually years. So my first patent, which I told you about the stapler, it took 9 years to grant. So even if it was a small concept, it took 9 years. So, but I had filed in the year 2011, which is long back. But now the process has become fast, and it depends upon your drafting quality, right? So the examination you have to clear. So these are the factors which are included. So around 4 to 5 years you can consider for granting of the patent. So it depends upon your draft and your innovation and your examination.
Okay, thank you. Any more question? Yes, okay. If no question, then I would like to thank Mrs. Ankita Nagarkar who accept our invitation. It is just interaction because up till now we have heard the lectures by PPT and all that, but she directly interact with us and explain about the patent filing, about there is no age, no education limit is there for filing the patent, and also about plagiarism and other research ethics. So all points she has discussed very well. So on behalf of our college, I would like to thank Mrs. Ankita Nagarkar. Thank you. Now this session is over. Now we have time of exam, that is we have MCQ test. This test start from this